Good morning. Good morning, esteemed faculty, distinguished guests, ladies and gentlemen. Welcome to European University of Cyprus School of Medicine and to the third day of the 12th International Biomedical Scientific Cyprus Congress. To begin, we would like now to call upon the chairs for the next session, oral presentation of selected abstract. Could Dr. George Lukates and Dr. Alpina Nikolusi please come to the stage for the presentation 1 through 4, followed by Dr. Katerina Minelau and Dr. Christina Kusparov for presentation 5 to 7.
Good morning. Hello, everybody. Welcome to our session. I would like to ask Dr. Excuse me, to find... Ναι, όχι, εντάξει, αλλά τον τίτλο ήθελα να βρω. Dr. Dr. Patsalis is from the Department of Molecular Genetics and Thalassemia from the Cyprus Institute of Neurology and Genetics, and he will present the title, a very interesting presentation. The title of his presentation is Preclinical Validation of CRISPR/CAS genome, editing approaches as advanced therapy for HBV IVSI thalassemia. Dr. Patsalis, the floor is yours.
Thank you. Good morning, everyone. Thank you for being here today. First, I would like to thank the scientific committee for the opportunity to present our work at this conference. So our research is focused on gene therapy for beta thalassemia, targeting the globally common and particularly in Cyprus IVS 110 missplicing mutation. The mutation caused the incorporation of 19 nucleotides from intron 1 into the mature mRNA, resulting in the production of a non-functional mRNA. While some functional beta-globin mRNA is produced, the majority of IVS 110 homozygotes have suffered from severe transfusion-dependent thalassemia major. Therefore, developing a mutation-specific therapy will be of significant social, economic, and commercial value. In the past, we have shown that targeted disruption of the IVS 110 aberrant regulatory elements using designing nucleases like TALES and CRISPR-Cas9 lead to genome modifications through non-homologous and joining repair pathway that are sufficient to disrupt the aberrant regulatory elements, prompting the spliceosome to utilize the normal acceptor site and therefore restore proper splicing. In 2022, we received national funding to validate our mutation-specific approach, including CRISPR-Cas9 TALENs and our recently published SPG7 adenine base editor. For ABE SPG7 base editor, we demonstrated effective T2C base edits at IVS 106, 08, and 09, restoring splicing in patient-derived hematopoietic stem and progenitor cells in vitro. We are comparing this with the FDA-approved SG1617 CRISPR-Cas9 strategy, targeting the BCL11A erythroid-enhanced element for the induction of fetal hemoglobin in the NBSGW immunodeficient mouse models. Today, I will present comparative data on IVS 110 CRISPR-Cas9, the SPG adenine base editor, and the SG1617 CRISPR-Cas9. Briefly, patient-derived hematopoietic stem cells were nucleoaffected with CRISPR-Cas9 as ribonucleoprotein complexes, while SPG7 adenine base editors and GFP were delivered as mRNA. After 2 days, the cells were divided for in vitro assessments for functional correction and of erythroid and myeloid lineage potential post-editing, and for in vivo evaluation of the long-term repopulating capacity of the edited hematopoietic stem cells in the bone marrow of primary and secondary NBSGW recipients 16 weeks post-transplantation. Starting with the assessment of genome editing efficiencies across individually nucleoaffected cultures and bulk input cultures used for xenotransplantation, graph A shows the IVS 110 CRISPR-Cas9 achieved high on-targeted disruption efficiency, reaching up to 85%, shown in blue bars, with no off-targets at the predicted RNF219AS1 site. The SG1617 CRISPR-Cas9 showed about 40 to 32% efficiency at the BC11A erythroid enhancer element in individual and bulk input culture, respectively. On graph B, analysis of the base editing efficiency of adenine base editor SPG7 showed high to T to C editing at the IBS 106, 08, and 09 sites, with a pattern indicating a shift from more frequent double and triple edits on day 6 to predominantly single edits per allele by day 9. Improvement of late-stage erythroid differentiation was measured at different time points during erythroid differentiation as the percentage of CD235 positive CD36 negative cells and as the percentage of enucleated cells in the CD235 positive population. Both analyses showed significant improvement of late-stage erythroid differentiation in IBS 110 CRISPR-Cas9 and SPG7 base edited cells, whereas the SG1617 genome edited cells showed marginal improvement only for enucleation relative to the untreated control. RNA analysis on day 11 of erythroid differentiation indicated that total beta globin expression levels shown in blue bars increased considerably in IBS 110 gRNA and ABSPG7 edited cells relative to the untreated, with splicing correction shown in stacked bars reaching up to 97% and 88% respectively, compared to the 67% in the untreated cells. In contrast, gamma globin expression in SG1617 edited cells dropped to 0.3 times relative to the untreated control. Analysis of the globin chains via reverse phase HPLC and different time points showed a significant increase of beta globin to alpha normal ratios to normal levels in IBS 110 CRISPR-Cas9 and ABSPG7 genome edited cell relative to the untreated control. Significant increase of gamma to alpha ratio was detected in SG1617 CRISPR-Cas9 genome edited cells relative to the untreated control. Clonogenic assays assessing erythroid and myeloid colony formation showed no significant difference between genome edited samples and the untreated control, suggesting that lineage potential remained unaffected by genome editing. Moving to the in vivo work on primary and secondary xenotransplanted NBSGW mice, we observed high bone marrow engraftment with 65% CD44 positive, 45 positive, 4.8% CD34 positive, and 3.8 for CD235 positive, while peripheral blood chimeras was at 6.5%. Statistical analysis showed no significant difference in the chimeras between genome-edited and untreated cells, indicating that genome editing did not affect long-term repopulating capacity. Statistical analysis of the multilineage potential of the edited chimeric bone marrow cells showed no significant difference in hematopoietic stem and progenitor cells, erythroid, B and T cells, and myeloid cells compared to the non-edited bone marrow controls, indicating that multilineage potential of the edited cells was unaffected. Comparison of genome editing levels of bulk input samples and primary recipient bone marrow cells showed approximately 50% decrease in the IBS 110 CRISPR-Cas9 bone marrow samples, similar levels for SG1617 CRISPR-Cas9, and surprisingly an increase by 20% for the SPG7 base editor. Targeted deep sequencing showed high on-target indel efficiency of 90% for IBS 110 CRISPR-Cas9, shown in blue, decreasing to 46 in the bone marrow in primary bone marrow cells, with consistent 8% of off-target effect at the RNF219AS1 shown in green. The SG1617 CRISPR-Cas9 had moderate on-target efficiency, shown in purple, of 60% dropping to 18% in the bone marrow, with no detectable off-targeting. ABSPG7 showed no intels at any site. Base editing analysis of the ABSPG7 showed high on-target efficiency in bulk hematopoietic stem cells, which increased by 30% in bone marrow, consistent with Sanger sequencing data shown before. In addition, we detected a shift of the base editing pattern from mainly single edits in bulk input hematopoietic stem cells to imbalanced distribution of single, double, and triple edits in primary bone marrow cells. The variation likely results from the extended exposure of base editor in the transplanted hematopoietic stem cell in vivo compared to the rapidly proliferating hematopoietic stem cells in vitro. To summarize, both tools, IVS 110 CRISPR-Cas9 and ABE-SPG7, outperformed the SG1617 CRISPR-Cas9 in editing efficiencies. All genome-edited cells maintain high long-term repopulating capacity, especially ABE-SPG7, which showed increased editing in vivo. Functional correction was highest for IVS 110 CRISPR-Cas9 and ABE-SPG7, while the multi-lineage potential of the edited hematopoietic stem cell was not affected. Off-target analysis showed minor off-targeting for IVS 110 CRISPR-Cas9 on RNF219-AS1 and undetected off-targeting on the SG1617 CRISPR-Cas9 or the ABE-SPG7. In conclusion, the IVS 110 specific tools outperformed the BCL11 specific CRISPR-Cas9, with ABE-SPG7 being the most efficient, safe, and promising for clinical use. However, further biosafety assessment is needed, including deep sequencing of the top predicted off-target sites and analysis of chromosomal rearrangements via CASSEC. Special thanks to our collaborators, advisors, and the thalassemia clinics in Cyprus for their support, and to all the patients and volunteers involved in the study. Thank you.
Thank you very much, Dr. Pacelli, for the very interesting presentation, and also thank you very much for keeping the time of the presentation.
So I was in time because the time
we have some time for one or two questions. Anybody from the audience? Any question? We don't have questions. Thank you very much. Thank you. And now we are calling Dr. Chrysanthi Avgoustou, who is from the Molecular Hematology Oncology Department of Karaiskakio Foundation. She will present a very interesting topic with the title of Investigating the Role of phosphorufructokinase platelet type in metastatic breast cancer dormancy. Thank you very much, Dr. Avgoustou. Please, the floor is yours.
Good morning, everyone. I'd like to begin by expressing how honored I am to be here and to have been selected to share my work. Today, I am going to present to you our project titled Investigating the Role of Phosphofructokinase Platelet Type, or PFKP, on metastatic breast cancer dormancy. According to the American Cancer Society, breast cancer comes first in estimated new cases in women during this year, and it is also the second highest in estimated new deaths. Invasion and metastasis is one of the 14 hallmarks of cancer, and it is responsible for the 90% of cancer mortality. During metastasis, cancer cells leave the primary tumor site and colonize into distant organs. This colonization does not happen by chance, but some organs have affinity to some others, and this is known as the seed and the soil theory. Dormancy is defined as the interval of time between the primary tumor treatment and metastasis to distal organs. During the dormancy period, cells remain in the G0 phase of the cell cycle and pause growth and any activity of an organism's life until their reactivation and metastatic outgrowth. As a consequence, these cells cannot be detected and therefore cannot be targeted by chemotherapy. This is a previous study performed by our team. In this experiment, metastatic breast cancer cells were intravenously injected in a mouse model. Bioluminescence experiment was performed to examine tumor growth each day. Cells were isolated on days 0, 20, 65, and 100, and RNA sequencing was performed to identify genes that could be involved in promoting or suppressing dormancy. One of the genes observed was PFKP. On day 0, PFKP levels were upregulated, where on day 20, levels were downregulated, and this indicates that PFKP is downregulated during dormancy period. Then on day 65, PFKP levels elevated again until the day 100, when the experiment was concluded. And this indicated that PFKP is upregulated in the escape from dormancy. PFKP is a protein coding gene, and the encoded enzyme controls glycolysis pathway by converting fructose 6-phosphate to fructose 1.6 biphosphate. Therefore, increased levels of PFKP induces high rates of cell proliferation. As I said before, based on the American Cancer Society, breast cancer has high mortality rates in women. Metastatic dormancy plays an important role in the recurrence of the disease, so understanding how PFKP is involved in dormancy can be helpful in the development of new therapeutic approaches. Based on this data, we hypothesized that increased levels of PFKP promote escape from dormancy by allowing adaptation to increased metabolic needs during cancer proliferation. And to evaluate this hypothesis, the effect of PFKP on breast cancer cell viability and proliferation in the presence and absence of glucose and oxygen were investigated, as well as the effect of lactate in breast cancer cell viability. To investigate the effect of PFKP, gene knockdown was performed using the inducible short hairpin RNA-mediated gene signaling mechanism. Tetracycline antibiotics like doxycycline are used as regulators of inducible gene expression systems. And two already established cell lines, MIV-LUC, SH-scrabbled, and SH-PFKP were used. So, when doxycycline is absent, the expression of shRNA is suppressed, leading to the expression of the target gene. And when doxycycline is added into the medium, shRNA expression is activated, leading to the knockdown of the target gene. This study began with a bioinformatic analysis using online databases to assess the PFKP expression levels in normal and tumor tissues. Then a real-time PCR was performed to confirm the knockdown of PFKP gene and also investigate the expression under specific condition, followed by an MTT cell viability assay to investigate breast cancer cell viability under the same conditions. Our bioinformatic analysis showed that PFKP expression may differ across the different stages of cancer progression, as shown in Figure A. And Figure B suggests that PFKP expression levels were elevated in breast cancer tissues compared to the normal ones. Our real-time PCR confirmed that PFKP was successfully suppressed following the knockdown. Gene expression was reduced in conditions lacking glucose compared to the controls, and this reduction in gene expression was more enhanced when glucose deprivation was combined with the knockdown of PFKP. MTT assay was performed in breast cancer cells under normoxia and hypoxia at 24 and 48 hours. Results have shown that under conditions, the absence of glucose reduced cell viability. This reduction was enhanced when combining glucose deprivation and PFKP knockdown by the addition of doxycycline in the medium. This assay was repeated, including the addition of lactate solution to investigate the possible effect of lactate on cell proliferation and viability under the same conditions. As on the previous experiment, lack of glucose reduced cell viability in both normoxic and hypoxic condition, and this was enhanced when lack of glucose was combined with PFKP knockdown. And what's interesting is that exogenous addition of lactate compensates glucose deprivation in both conditions, leading to increased cell viability. Cancer cells rely on glycolysis for energy production, a phenomenon called the Warburg effect. When glucose is absent, cells are unable to do so. So by supplementing them with the lactate can be taken up by the cells, converted into pyruvate, which can be used as a fuel for TCA cycle for energy production. In an organism, cancer cells support one another by sharing metabolic processes, and this creates a synergy and enabling cancer cells to thrive in different environments. This is called the lactate shuttle. Our conclusions suggest that glucose starvation reduced breast cancer cell viability both under normoxic and hypox conditions. When glucose starvation is combined with PFKP knockdown, this reduction is even more enhanced. Glucose deprivation is rescued when lactate is exogenously added, and lactate can now be recognized as a fuel for TCA cycle and as a pro-tumorigenic molecule. Future directions include in vivo studies with PFKP inhibitors, combination of PFKP inhibitors with other therapeutic agents such as chemotherapy, and combination therapy with glycolysis inhibitors. Before I conclude, I would like to take a moment to thank those who have supported and contributed to this project, including my supervisors, Panagiotis Papagiorgis and Dr. Christiana Neofidou. Also, a big thank you to Dr. Paul Kosteas, the team at the Center of the Study of Hematological Malignancies, and the team of the Molecular Pathology and Oncology Department at the Garskage Foundation for their invaluable guidance and support. Thank you, everyone, for your attention.
Thank you very much. Thank you very much, Dr. Avousti, and thank you for keeping the time as well. I think we have some time for one question. Any question from the audience? Okay. Thank you very much, Doctor. There is a question? No. Okay. Thank you very much, Dr. Avousti.
We can call Dr. Athanasios Metaxas from the Department of Neurobiology, Institute of Molecular Medicine, University of Southern Denmark.
Yes, that is another affiliation that I hold, also along with my affiliation in EUC.
Okay. And the title is Serotonin 2B Receptor Expression and Binding the Brain of APPSWE /PSIDN9 transgenic mice and in Alzheimer's disease brain tissue.
Exactly.
Thank you.
So my title today is about the involvement of serotonin 2B receptors in Alzheimer's disease, and we've started that using a mouse model, the APPS1, and also post-mortem AD tissue. Alzheimer's disease was first described in 1906 by Dr. Alois Alzheimer, who was caring for a period of 5 years for patient 0 of Alzheimer's disease, Augustine Dieter. And he described the cardinal symptoms, clinical and neuropathological, that characterize Alzheimer's disease. And this involve both a cognitive impairment, progressive, but also non-cognitive impairment. And today we know that non-cognitive impairment in Alzheimer's is mediated by the serotonergic system, right? This is a schematic of the origins and the projections of the serotonin system in the human brain. And as you can see, the serotonergic somata are situated in the brainstem, projecting elongated axons that reach throughout the brain. A characteristic feature of serotonergic transmission is that serotonin is released by both synaptic and extrasynaptic mechanisms, so it can reach not only the postsynaptic neuron, but also astrocytes, microglia, and other cell types. Serotonin exerts its effects via at least 14 receptors in the human brain, organized in 7 big receptor families. And my talk today is about the 5-HT2B receptor, which is a G-protein coupled receptor acting through PLC secondary systems. What do we know about serotonin in AD? I've already mentioned that the non-cognitive symptoms, depression, apathy, all of this stuff are mediated by serotonin, but we also see genetic polymorphisms in some receptor genes that increase the risk of AD. We see serotonergic neural loss by post-mortem studies and also by in vivo PET imaging. We see reduced serotonin levels in AD and changes in a variety of serotonin receptors. This is a cartoon from 2023 describing all the changes in serotonin receptors that we see in the human brain up until last year. And what you can see here is that serotonin 2B receptors are missing from this schematic. There is a good reason for this. The 5-HT2B receptor was the last receptor to be cloned from the serotonergic family in 1994 in the human brain. And 4 years later, in 1998, it was already discovered that when you have drugs that are agonists at the 5-HT2B receptor, you develop a characteristic pattern of heart failure, which involves the destruction of cardiac valves. So it's actually a known target for the pharmaceutical industry, and nobody was paying attention to this receptor up until approximately 5 years ago, where we got the crystal structure of the 5-HT2B receptor, and then some studies have been coming out regarding the role of 5-HT2B receptors in the CNS. So we now know that 5-HT2B receptors play a role in SSRI antidepressant function. So if you knock out the 5-HT2B receptor in a mouse model, you don't get antidepressing effects from your SSRIs. There is a role in drug reinforcement, so psychostimulants like cocaine act via the 5-HT2B receptor. There's a role in schizophrenia, and particularly as far as neurodegenerative conditions are concerned, there is a role in motor neuron disease. So based on this, and based on our interest in the interactions between serotonin and Alzheimer's, what we did is actually a very basic study examining the regulation of 5-HT2B receptor mRNA expression and protein binding in a transgenic mouse model of Alzheimer's disease and in post-mortem tissue. The hypothesis, of course, being that the 5-HT2B receptor would be involved in the context of AD. We use this mouse model of Alzheimer's disease at different time points, which represents different stages of AD pathology. We used human female control at AD confirmed post-mortem tissue. We measured mRNA expression by RT-qPCR and protein levels by binding and not by using antibody-based techniques, because in our experience we could not either develop or find 5-HT2B selective antibody. So what I'm showing here is 5-HT2B mRNA expression in the entire brain tissue, so isolated mRNA from the entire brain tissue. This is relative fold expression relative to 3-month-old wild-type mice. And the first observation we have is that in the whole brain, 5-HT2B receptors, the expression is progressively increased with age in the wild-type mice. This regulation is not observed in the transgenic mice, and we also get a reduction at aged mice here by 18 months, more than 18 months of age. So 5-HT2B receptors at the whole brain levels are reduced in transgenic mice, but Alzheimer's disease does not affect the entire brain like uniformly. It specifically affects neocortex and the hippocampus. So when we measured the expression in the neocortex and the hippocampus specifically, we see that in aged transgenic mice, there is actually a doubling and increase in the expression of 5-HT2B receptors relative to control, something that we also observe in the 20-month-old hippocampus. We managed to replicate this increase in the neocortex using human post-mortem tissue. So in the frontal gyrus of confirmed AD patients, there is increased 5-HT2B mRNA expression compared to age-matched controls. And because there is evidence that say that the microglia are the main cell type that express 5-HT2B receptors in the mammalian brain, we also confirmed this finding by showing in different experiments that when you isolate microglia, you get an increase of 5-HT2B receptor in microglia compared to other cell types. Regarding the binding levels, the conclusion here is that the protein levels of 5-HT2B receptors in the brain are low. We could observe protein binding after 3 months of age, so starting at 6 months. In both the neocortex and the hippocampus, there is a tendency, statistically significant, of reduced 5-HT2B receptor binding in the brain. However, we need to boost up the N numbers to get age-specific significance. These are representative autoradiograms in sagittal brain tissue from transgenic and wild-type mice, from AD and control patients, showing how little the binding really is. And these are coronal heart tissues from a wild-type mouse, which show, again, that the receptor is highly expressed in the heart. So we use this as a positive control. So our conclusions are that 5-HT2B receptors are constitutionally expressed in both the mouse and the human brain. The levels are low. Whether you measure them by PCR, you get the signal around 30-31 cycles, or you measure the protein. 5-HT2B mRNA levels are increased in the neocortex of H transgenic mice and in AD subjects, and the binding levels are overall decreased in the neocortex and the hippocampus in transgenic mice compared to control. We couldn't identify protein in the human brain. The levels were very low. I would like to thank my PhD student Marco and all of my collaborators and you for your attention. Thank you.
Thank you very much, and keeping the time as well. So we still have time for a question. Anybody? Okay, thank you. Thank you very much. We can move now and call Dr. Arman Kalsahagian from the Department of Life Sciences of School of Life and Health Science of University of Nicosia with a presentation on SSVEP and native gamma responses to phase reversing and static visual stimuli in healthy individuals.
Thank you for being here. My name is Arman Kalsakian. I'm currently a neuroscience student at the Cyprus Institute of Neurology and Genetics. Before this, I was studying human biology at the University of Nicosia, and this was my bachelor's thesis under Professor CYPRUS. So the idea of this experiment is to record certain responses in the brain, the SSVP and native gamma responses, to specific visual stimuli. So I'll begin by introducing the topic, briefly explaining the methodology, moving on to the results, and then the future direction and conclusion. Imagine, by a simple pattern, or grating as we call it, you could improve brain health. Or you can reduce the progression of certain neurodegenerative disease. And in this case, Alzheimer's disease. Alzheimer's disease, for those who don't know it, is a neurodegenerative disease where neurons die. And this happens due to many reasons. But the main theory is that you have extracellular amyloid plaques, and those plaques lead to a cascade of events, tau tangles, and neuron death. And if you look at an Alzheimer brain, you have atrophy. And usually you treat the symptoms of Alzheimer's, but not the disease itself. And some drugs have came out recently trying to treat the disease, but they have a lot of side effects and not as effective. So of course we want new therapeutic approaches. And this is a therapeutic approach with many scientists around the world, and it's a pretty niche field. So the idea is that scientists realized there were weaker gamma power in the gamma oscillations in the brain in Alzheimer's disease patients and in mild cognitively impaired patients. And there's research showing gamma's role in cognition. So the idea was that perhaps with an external stimulator, we can stimulate the brain at the gamma frequency range and strengthen those networks and possibly improve cognition. And one way you can do this is if you have a phase reversing grating. So essentially you have black and white patterns, and those black and white switch back and forth 20 times a second, and you get a response in the brain at 20 hertz, but also at its harmonic at 40 hertz. And there has been studies in mice models of Alzheimer's and in a few studies in humans. Gamma was entrained, so the intrinsic oscillations were entrained to this external stimulation. There were cellular and molecular changes in mice. There was reduced progression in some studies of the disease, and there was less brain atrophy, but of course it's not that simple. Other studies went against this, so they showed no gamma entrainment and little to no cognitive benefits. And this can be due to a lack of normalization, different protocol uses, different analysis methods between teams. And that's why we wanted to investigate this our way. So firstly, we want to investigate if we get native gamma responses, SSVP or both, to a phase reversing grating that I showed you, or what responses we would get to a static grating. Static meaning you show the visual stimulation of the pattern for around 4 seconds, let's say. You do this many times. And we wanted to see the propagation of this response to structures in the brain, specifically the medial temporal lobe. But due to limitations of my bachelor thesis, time and resource limitations, we couldn't do the second part yet. And just to briefly go over what those responses are, to know some theory behind it. When you show a visual grating, it goes through your visual pathway, it reaches your occipital lobe, and you get a response in the visual cortex, which can be recorded with an electrode, EEG. And you do this over many trials, and you get waveforms, right? When you average those waveforms, you get the evoked response. Because the evoked response is time-locked, so it happens at a specific time after the visual stimulation presentation, but it is also phase-locked, meaning the phase of the response is always the same. So when you average it, everything else cancels out, and you get the evoked response. So the same thing is true for the steady-state visually evoked potential response. It's just a visually evoked response to the phase reversing grading. So you'd get 20 waves a second when you average the trials. Now, the induced response is a bit more complicated. But simply, it is time-locked, so it happens at a certain time after the visual stimulus presentation, but it is not phase-locked. And this has to do with the properties of systems, and it has to do with higher levels of processing. Simply, the methodology, it was a neurocognitive experiment. You had the task, the event, you had the EEG system, and we recorded it using a laptop, and amplifier and a trigger system. This was what the participants saw during the experiment. This was the protocol. It lasted around 25 to 30 minutes, and you randomized this. And there were 4 participants due to the pilot study nature, so it's a prerequisite study to follow-up studies. I was one of the participants. I was more excited than I seem in this photo. And this was another participant, and this is how the experiment was. And briefly over the results. So we did get around the 20 hertz SSVEP response. And we didn't get a strong harmonic response at the 40 hertz, but we're going to investigate this. We had an induced response at 20 and 40 hertz, but the y-axis is times 10 to the power of 4. So it's exaggerated and it's inconclusive. And there was strong phase lock. The phase locking value was high, meaning the phase we got from the evoked response for each trial was it was very strong, which shows strong evoked response. Now, there was no evoked response to the static, which is unexpected. We expect to get the static evoked response. That could be due to many reasons we won't cover right now, and we can't really conclude the induced response. But please, don't be discouraged. This happened in a span of 2 weeks due to the limitations that we had, and it's kind of a setup study for the actual study that we're going to do. We had the 20 hertz response, but a small peak at the 40 hertz. Stimulus tolerance was lower than expected, and of course we have to look into this because it is a form of therapy that we're promoting here. Again, static did not produce the evoked response, but we will look into that. Now, the most important thing is the future direction of the study. We are working on, is fine-tuning the setup and the analysis pipeline. We are going to conduct a follow-up study soon on healthy individuals, 20 to 30 healthy individuals, also investigating the second part, which was the propagation of the response. We want to do statistical analysis as well. And depending on the results, we will move on to Alzheimer's patients, where we can see if the responses we got can translate into a therapeutic value. And a thing I want to introduce real quick is personalized visual stimulation. Perhaps in the future, based on specific parameters, you can characterize certain visual patterns to fit each patient. So this lies in the field of precision medicine. And to conclude, you're probably not going to remember most of what I said, but just the take-home message is that this method is non-invasive, so it can be done at home just by a video, essentially. And it is a visual gamma stimulation therapy method. It's pretty new. In the last decade, there's been some investigation into this. And it can potentially reduce the progression or ameliorate Alzheimer's disease. Thank you for your time.
Thank you again for respecting the time. Do we have any questions? Okay. Thank you very much.
Thank you. Yes. Yes. No problem. It's all okay. Just to say nice good morning to distinguished chairpersons and to all the colleagues, and to say my thanks to Professor Patrikios for inviting me again to be here with you for this very nice and important Congress. My presentation today
Just if you can give us a minute. First of all, thank you very much for accepting to attend this conference. My colleague Katerina Melelau will just give us one or two details about you and your talk. Thank you.
Yes.
So, Professor Andrea Stanimirovic is from the University of Applied Health Sciences in Zagreb, Croatia, and also the School of Medicine at the European University of Cyprus. And the title of the presentation is New Anti-Aging Strategies Review.
Okay.
The floor is yours.
Thank you very much for introducing me. And I would like to point out two things at the beginning of this lecture. This lecture is not scientific lecture which represents scientific investigations like the lectures before. It's just a short introductory review of one new discipline in the medicine which has the name longevity medicine or anti-aging medicine. It's a very new discipline which is established nowadays in the last few years, primarily in the United States, but nowadays also in Europe. And its task is to eliminate, reversing or reducing the effects of aging. And I would like to grow your interest to include in this part of medicine, because the aging is, as we know, the major risk for all the chronic diseases like cancer, cardiovascular disease, neurodegenerative diseases, and diabetes, of course. And it's very important that World Health Organization recognizes 10 years ago the aging as a disease, no, not as a physiological process, as a disease. And these facts simulated the research on aging and aging-related diseases. And you see that anti-aging market in the drugs and the repurposing drugs is going increasingly up. And you see in 5 or 6 years, it will double, especially in the States. So, about the process of aging, of course, what we investigate in the process of aging are the hallmarks of the aging, which have, of course, and they must have a causal role in aging, like genomic instability, telomeres shortening, mitochondrial dysfunction, senescent cells, stem cells, senescent cells, dysbiosis, and many other hallmarks. So, the main task of the longevity medicine is not primarily to prolong the lifespan, but to prolong the health span in the lifespan. So standard medicine succeeded to prolong lifespan up to 80 or 85 in majority of the countries, but the last 10 years of the life of every person is plagued with chronic diseases. So the task of the anti-aging medicine is to prolong this pre-diseases healthy aging period. First attempts were made, of course, with the human growth hormone and estrogen, and they were promising, but of course, these two drugs, when repurposed to anti-aging, has the main problem with the decay introduced and developed and being included in the developing of carcinogenesis of various tumors, so they were not very acceptable. So, first attempts were followed by calorie restrictions, measures, and fasting. You know intermittent fasting, which is very popular nowadays in Western world, but intermittent fasting and calorie restrictions somehow is very challenging and has its potentially adverse effects. So
they developed, the scientists, the fasting mimicking diets, and we must point out the one attempt to mimicking fast calorie restriction diets with ketone bodies, with introducing into the therapy the beta-hydroxybutyrate. It's a ketone body, and it promotes ketogenesis, which means that the body is forced to use fat instead of the sugar for energy. We call this ketogenic diet. So when we use beta-hydroxybutyrate as a ketogenic body, it's called anti-aging ketone body. It's very important to see this last sentence, that it's extend mean lifespan by approximately 20%. Of course, it is for the animal studies for the time being, because the human studies are still in process. And you see that these receptors, peroxisome, presented activated receptors alpha in the brain, which are responsible for the levels of triglycerides and the homeostasis in the organism, are very important in this process of ketogenesis. So you see also this slide, and you see this slide, which would like to understand, to make clear this process. So the Buck Institute in California, which is the first institute in the world which promotes aging research, made up to now many mice studies with ketogenic diet and with very good results. And of course, they started last year the so-called BIKE study, Buck Institute Ketone Ester Pilot Study, and it's launched first human clinical trial, and we will see how this will end up. This will last for 2 years. Another drug is metformin, which is, very old drug for diabetes, especially for diabetes type 2, and very popular drug, and the results of anti-aging effects of metformin are very, very promising, because it activates AMP-activated protein kinase and inhibits mammalian digit of rapamycin signaling pathways. They are included, of course, in the aging hallmarks. It reduces oxidative damage, promotes positive stress, promotes autophagy, which is very important as anti-aging process, and apoptosis. And all these processes are, in fact, the elimination of damaged cell components, which leads to improved cellular health. So first it was used, and this is nowadays used for treating diabetes, but not only for diabetes. We have very good results with repurposing metformin for cardiovascular diseases, because it reduces cardiovascular mortality, all-cause mortality, by the incidence of cardiovascular events. You see these molecular effects on vascular endothelial cells, smooth muscle cells, and it works against the chronic systemic inflammation, improves the function of vascular smooth muscle. Also, it has results in musculoskeletal diseases, neurodegenerative diseases. It has potential anti-cancer properties, especially in pancreatic, breast, colon cancers, because it has some effects on preventing the genomic instability and cell proliferations and angiogenesis in the cancers. And as a dermatologist, I have to point out a little bit of the skin, that it has very good results on the skin, metformin by these cells. We must take care of the side effects, especially of lactic acidosis and vitamin B12 deficiency, but we can easily cope these side effects with another drugs or with particular care. Also is in the process the TAMI, target engaging with metformin study, which is now in process in Buck Institute, and we shall see the results of this study very soon. Very important, another repurposing drug is rapamycin as an anti-aging drug. Is in fact macrolide, a kind of antibiotic from bacterium Streptomyces hygroscopicus, but it has also immunosuppressant effects, and it is used like anti-cancer, like cytostatic drugs. It binds to the enzyme called mammalian target of rapamycin, and it allows autophagy to proceed in way to mimic calorie restriction. Very important. It mimics calorie restriction, so you don't need to have fasting or fasting mimicking diets, but to take rapamycin, which in fact eliminates hyperimmunity rather than suppresses immunity, but in any age, because calorie restriction doesn't work in old age, only in the early age, but the rapamycin inhibits this mammalian target of rapamycin C1 at any age, so it extends lifespan, whether started late or early in life on the mice studies. very good results in cardiovascular system, in the nervous system also, delays the onset of neurodegenerative diseases such as Alzheimer's, Parkinson's, and Huntington's, and it has very good effect on musculoskeletal system by improves bone health by delaying bone loss and protecting osteoporosis. Also, to point out the skin as a dermatologist, beneficial effects on various skin aging-related conditions, including wrinkles, pigmentation, and loss of elasticity. So, side effects we have, of course, like as every drug, like stomatitis, mucositis, non-infectious pneumonitis, hyperglycemia, so in higher dosage, it slows cell proliferation, which can result in thrombocytopenia or anemia. So we must measure and we must titrate the dosage of rapamycin very, very carefully. And blood levels should be measured because its concentration varies among individuals, so it's a challenging task to make the optimal dosage of rapamycin per individual, per each person. But luckily, scientists started to combine treatment of metformin and rapamycin drugs because we can use the good sides of both metformin and rapamycin on each other to eliminate the bad side effects, so it could protect a new repurposing new combined therapy. As we see, they can boost each other's effectiveness and block side effects, as it said in this slide, and some studies also are in current process to see how they are working together. One of the new drugs also repurposing, not old, but not new, but old drug for hypertension is rimeldinin, which has also this very interestingly possesses two effective but activators of AMP protein kinases and inhibition of mammalian target of rapamycin, and it gets very good results. And for the end, we also must mention the rosmarinic acid and glycation, because the glycation is one hallmark of the aging, and rosmarinic acid as a plant drug has anti-glycation properties. This is a short overview of the time being very popular anti-aging strategies, just to show you the myriad of the scientific proveness. You see
Professor Stamirovic, without wanting to disturb you, please, you need to wrap up because we are
yes,
already
now we are over. And just to conclude that at the end, with limitations, human trials are still in process, so we must be very careful to avoid the risks and side effects, and it's crucial to continue exploring new approaches. But I think personally, and I would like to grow your interest, that the longevity is very expecting new discipline. Thank you very much for your attention.
Thank you very much on an extremely important and interesting topic of anti-aging. If you can find, if you
later on the coffee.
Absolutely, thank you so much.
Thank you.
We now need to move on to Dr. Fotini Boya from the European University Research Center, who will be giving the presentation on behalf of her team. The title of presentation is The Synergistic Effects of BET Inhibitors and Immune Checkpoint Blockers in Pancreatic Cancer Treatment. Fotini, the floor is yours.
Thank you very much. Good morning, everyone. First, I would like to thank the scientific committee for giving me the opportunity to present my PhD work about the synergistic effects of BET inhibitors and immune checkpoint blockers in pancreatic cancer treatment. Based on 2024 statistics, pancreatic cancer remains the fourth leading cause of cancer-related death in men and the third in women. With a persistently escalating incidence and minimal change in mortality rates, pancreatic cancer is predicted to become the second cause of cancer death within the next 6 years. The average lifetime risk of developing pancreatic cancer is around 1.5%, which translates to 1 in 64 people. The aggressive behavior and the rapid development of metastasis explains why pancreatic cancer patients have a 5-year overall survival rate of less than 13%. Moreover, pancreatic cancer symptoms such as type 2 diabetes outbreak, abdominal and back discomfort, and weight loss are not specific and are often misinterpreted, leading to poor and late prognosis. There are many reasons for the increased mortality rates of pancreatic cancer. First, pancreatic cancer has limited therapeutic options, including surgery only when the tumor is resectable and chemotherapy using FOLFIRINOX. Despite the success of immune checkpoint inhibitors in other tumor types, pancreatic cancers remain unresponsive as they are amongst the most immune-resistant tumor types with highly desmoplastic and immunologically cold tumor microenvironment, leading to low proportion of cytotoxic immune cells and increased presence of tumor-promoting Tregs and myeloid-derived cells. Therefore, inefficient cytotoxic drug delivery can dramatically reduce therapeutic efficacy and thus negatively affect the quality of life and survival of pancreatic cancer patients. On a different note, bromon domain and extraterminal domain proteins can specifically bind acetylated lysine residues in histones to regulate gene expression. BET proteins have been found to be involved in the oncogenesis of various cancer types. Overexpression of BRD4 is found in pancreatic cancer, and its targeted reduction leads to inhibition of tumor growth. Based on these findings, researchers have initially developed various first-generation BET inhibitors, which compete with the acetyl-binding pockets of the proteins to inhibit their functions. These type of inhibitors are both tested preclinically and clinically as promising approaches to target both hematological and solid malignancies. Recently, second-generation BRD4 targeting protein degraders, which are more stable and have a more rapid and sustained action, like the MZ1, were developed. Studies using both first and second-generation BET inhibitors have shown great promise by inducing tumor-suppressive activities, including suppression of oncogenes expression, reduction of desmoplasia, remodeling of tumor microenvironment, and enhancement of anti-tumor immunity. Studies by our group and others have shown that repurposing of antifibrotic drugs, such as tranilast, which is approved in Japan and South Korea, has demonstrated the ability to enhance drug delivery and efficacy of chemotherapy, nanotherapy, and immunotherapy in breast cancer. Therefore, our overall hypothesis is that combined treatment of pancreatic cancer with tranilast along with MZ1 may act synergistically to remodel the tumor microenvironment towards improved anti-tumor immunity to enhance the efficacy of immunotherapy. First, to identify the most effective immune checkpoint inhibitors to be used in this study, we developed two syngeneic mouse models for pancreatic cancer and evaluated the efficacy of anti-PD-1, anti-4-1BB, and anti-LAG3 monotherapies to suppress tumor growth and promote anti-tumor immunity. We found that anti-PD-1 and anti-4-1BB monotherapies promote anti-tumor immunity without affecting primary tumor growth in the PANGO2 mouse model, whereas there was a statistically significant delay in the KPC model. Our flow cytometry data suggests that both anti-PD-1 and anti-4-1BB significantly increase the percentage of cytotoxic T lymphocytes, whereas they decrease the percentage of Tregs as well as the ratio of Tregs to cytotoxic T lymphocytes. Since these two monotherapies were found to be the most effective, we decided to use them as cocktail ICMBs during various treatment combinations with tranilast and MZ1. Our data indicated that only MZ1 was able to enhance the efficacy of immunotherapy, first by delaying the primary tumor growth, as indicated by the graph and the bioluminescence images, whereas surprisingly, the triple combination with tranilast, MZ1, and ICMBs was not as effective. At the end of the experiment, tumors were excised and sent for single-cell RNA sequencing. The analysis revealed 14 distinct cell populations, clusters including 8 pancreatic cancer population, 3 myofibroblastic cancer-associated fibroblasts, 1 inflammatory cancer-associated fibroblast population, and 2 immune cell populations. Quantification of the percentage of each cell cluster in the different treatment groups indicated decrease in 3 PDAC pancreatic cancer subpopulations, increase in the M1 type macrophages in the myofibroblastic calves, and the CD4 positive T cells in the combination treatment of MZ1 plus ICMBs. To independently validate our single-cell RNA data, we performed spectral flow cytometry analysis. This method allowed for the identification and visualization of 20 distinct T lymphocyte clusters according to their specific marker expression profiles. The data presented here in the figure represent the T-SNP plots of the identified T lymphocyte cluster in the different treatment groups. Interestingly, the analysis revealed increase in CD8 positive and CD4 positive effector memory T cells in the ICMB and the combination treatment of MZ1 plus ICMB. Our preliminary analysis in the myeloid cluster indicated that M1 tumor-associated macrophages were increased, the M2 tumor-associated macrophages were decreased, and the ratio of M1 to M2 tumor-associated macrophages was increased in the ICMBs and the combination treatment of MZ1 plus ICMBs. Finally, to evaluate the effect of the different treatments on the animal overall survival, we performed a survival study. Kaplan-Meier survival curves relieved a significant improvement in the overall survival in the ICMB and the ICMB plus MZ1 treatment groups. To conclude, monotherapies using anti-PD-1 and anti-4-1BB ICMBs were effective in inducing anti-tumor immune responses, whereas there was no effect using the anti-LAG3 ICMB. Some of the PDAC subpopulations were decreased in the MZ1 plus ICMB groups, which was in agreement with the observed decrease in the tumor growth. Increase in infiltration of cytotoxic CD4 positive T cells and anti-tumor M1 type macrophages, as well as increased percentage of myofibroblastic CAFs was observed in the MZ1 plus ICMBs group. And finally, the ratio of M1 to M2 macrophages was significantly increased in the MZ1 plus ICMBs group. Our ongoing work includes an in-depth analysis of single-cell RNA sequencing data to reveal gene expression signatures associated with specific cancer and immune cell population that could explain the responses in combination immunotherapy. Further analysis of the spectral flow cytometry data to reveal possible changes in specific immune cell populations in the T cell or the myeloid panel that may explain the responses to the MZ1 plus ICMB treatment group. And as a more long-term goal, we aim to develop nanoformulations carrying PROTAC BET inhibitors to improve their delivery and efficacy in pancreatic tumors while minimizing the possible side effects and improving the efficacy of immunotherapy in pancreatic cancer. This work could not be performed without the generous support by the Research and Innovation Foundation through the Excellence Hub project coordinated by Professor Papagiorgis at European University Cyprus. I would also like to thank my PhD supervisors, Professor Papagiorgis and Dr. Christodoulou, and our collaborators at the University of Cyprus, the Center for the Study of Hematological Malignancies, and the George Washington University. Thank you for your attention.
Thank you very much, Fotini. We have time for a brief question. No, from my side, I would like to thank you on behalf of the organizers for presenting our work, and congratulations to you and all your collaborators team.
Thank you very much.
Thank you, Fotini.
Okay, so next we have Professor Zaravinos from the Department of Life Sciences at the School of Sciences at the European University of Cyprus. The title of the presentation is Multiomics Analysis of an Exhausted T Cells Molecular Signature in Pan-Cancer.
Apologies, but your name is?
My name is Chris Rigopoulos, and I am a fourth year undergraduate biology student of Professor Zaravinos.
Okay.
And I will give the talk on behalf of
Okay, so there must have been a mistake in the program because we thought, but we welcome you as well, just the same.
Thank you. Hello everyone, and thank you so much for this opportunity, especially to my supervisor, Professor Zaravinos, and Professor Ioannis Patrikios. My name is, as I said, is Christos Grigopoulos, and I am a fourth year undergraduate biology student. And today I will give you a talk on multiomics analysis of exhausted T cell molecular signature in pan-cancer. Throughout this presentation, we will explore how multiomics approaches allows us to examine the complexity of exhausted T cells in cancer. To understand the complexity of exhausted T cells in cancer, let's first recognize the intricate landscape of cancer biology itself. Cancer isn't just a single disease, but rather a spectrum characterized by remarkable complexity. We are dealing with a combination of genetics and transcriptional changes, diverse phenotypic traits, and significant histologic variations. What is truly challenging is that this heterogeneity can exist not only across different patient's tumor, but also within a single patient's tumor, adding layers of difficulty in our quest for precise treatment options. The recent revisions on hormones of cancer, as outlined by Let's Go, bring to light the dynamic interplays between cancer-specific attributes and especially what we will investigate, avoiding immune destruction. Regarding the relationship between cancer and immune system, T cells are key players in our immune system. They normally help our body detect and destroy abnormal cells, like those in cancer. But sometimes, due to persistent exposure in antigens in chronic diseases like cancer, T cells lose their effectiveness and enter a dysfunctional state called exhaustion. So these T cells are called exhausted T cells. And they are characterized by an increased expression of inhibitory receptors like PD-1, LAG3, TIM3, CTLA4 and TIGIT, and a combined decreased production of essential cytokines like interleukin-2, interferon gamma, TNFA, and granzyme B. Overall, these lead to decreased cytotoxicity, decreased proliferation capacity, and impair anti-tumor functions. By studying these markers that identify these exhausted T cells, we hope to understand why these cells stop working properly and how we might help them reinvigorate and fight cancer better. The aim of the study is to identify key markers and patterns, to link the characteristics of T exhausted cells to patient outcomes, to explore potential therapeutic targets, and in general contribute to precision medicine. Regarding the materials and methods, we've used 6 specific T-exhausted markers: TIGIT, HALCR2, PDCD1, LAME, LAG3, and CXCL13. And the workflow of the study is also shown here. We extracted the signature from the TCPA database, and we collected our data from 33 cancer databases using the main TCGA database. So we investigated this signature in terms of mutation, expression, methylation, immune infiltration, and drug sensitivity. Regarding our findings, firstly we focused on the differential expression of our signature. One of the first things that we found was that in kidney renal clear carcinoma, all 6 genes were overexpressed, as we can see here from the box plot. We also saw that in thyroid carcinoma and chromophobe kidney cancer, LAME gene was underexpressed, indicating that probably has a more specific role in those 2 specific cancers. We then investigated the survival of our patients using PrognoScan. We extracted the Kaplan-Meier's curve below, showing the survival between low expression groups and high expression groups. We found that low expression groups in uveal melanoma, Kaplan-Meier 1 and 3, were associated with better survival rate, while Kaplan-Meier's 2 and 4, regarding skin melanoma and breast cancer, low expression groups showed worse survival, indicating that this signature may function as tumor suppressant in those 2 cancer types. Then we investigated the mechanism and the pathways that these genes regulate. And one of the biggest surprises for us was the role of LAME gene in apoptosis. Usually, the majority of T-exhausted genes promote apoptosis, as it is the essential way to kill off damaged cells. But in this case, LAME does the opposite thing. It stops apoptosis from happening, and by blocking cell death, we hypothesize that LAME could be giving cancer cell a survival advantage. We furthermore sought to explore the potential correlation between our gene expression and immune system, especially in bladder cancer, where we found high level of immune infiltration score and correlations with the majority of immune cells. This means that even though, as we said, the T cells are exhausted, the immune system is still trying to find a way, recruiting the other immune cells to fight the cancer. This type of cancer, bladder cancer, might respond better to treatments that target these exhausted T cells and reactivate them. At the same time, we attempted to explore the mutational rate of these T-exhausted signature genes in pan cancer. Our results indicate that in uterine corpus endometrial carcinoma and skin melanoma, these two cancers harbor the majority of the mutations, but this is something that we were expecting because these two cancers are generally highly mutated. And we also observed that the majority of these mutations were missense mutations of the C2T class. Then we focused on finding any possible correlation between the methylation of the genes and the tumor burden. We found that LANE was highly methylated in the majority of cancer types, while the other genes showed a reverse effect, demonstrating lower methylation levels. And finally, we explored possible drugs that could be used in order to treat malignancies. We observed that the majority of those genes were negatively correlated with the drugs, implicating that patients who have a low expression of these genes may have an increased drug resistance. We are aware of the fact that we lack in vitro validation, but nevertheless, I think that our research offers new insights into the molecular signature of exhausted T cells across diverse cancer types, like the overexpression of T-exhausted genes, especially in kidney, renal cell clear carcinoma, the fact that the apoptosis pathway was the most inducible of all the paths, the fact that the T-exhausted markers are not significantly mutated across different tumors, with the exception of uterine corpus endometrial carcinoma and kidney melanoma. LANE was hypermethylated across all cancer types, in contrast with the other genes. And we also think that we contributed to the corroboration of TIGIT's role as an immune checkpoint inhibitor therapeutic target. And these findings not only deepen our understanding of cancer immunology, but also open pathways for more targeted and personalized cancer therapies. And before ending, I would like to thank once more my supervisor, Professor Apostolos Zaravinos, and Dr. Elias Georgakopoulos Soares, who helped me with this study. Thank you for your attention.
We have time for a quick comment or question. No? In that case, I would like to thank you for a very comprehensive presentation and very schematic and visual representation of your results as well. Thank you.
At this time, we invite the chairs, Dr. Anastasia Stefanou and Dr. Christina Gospari, to the stage.
Okay, so I'd like to introduce our next speaker, Dr. Nikolaos Papaioannou, a resident doctor at the Asclepios Voulas at the Department of Internal Medicine. And the title of his presentation is Arterial Hypertension and Microvascular Disease.
Good morning, thank you, Mr. President. And therefore, may I want to thank you, the committee, for the very kind invitation, especially Professor Katridios, for having me here. And let's talk about our issue, which is arterial hypertension and microvascular disease. And we know that microvascular disease is a manifestation and disease which comes from ischemia in the microvascular coronary artery bed. It refers to vessels that are below 500 micrometers, and when we try to check the pathophysiological mechanisms, we're going to see that we may have a spasm in the microcirculation, endothelial dysfunction, remodeling, or even perivascular fibrosis. As you can see in this amazing graph, we know that it's a disease that the latest years has come up and we start dealing with it. It's true that when we have a patient with angina and we undergo a coronariography invasive in this patient. Do you know that in a percentage still 70% we found no obstruction, which means that only 30% of our patients with angina present with obstructive coronary artery disease. As you can see so here, when we have a patient with obstructive coronary vessels, it's only in the peak of the ass mountain, while on the bottom you can see that there are so many other factors that can affect the coronary tree. And it's true. During an invasive coronariography, we can only detect the 5% of the total coronary tree. The left 95%, we can't see it, we can't estimate it, we can't evaluate it, and that's why it's a disease with unknown till now for us. And we try to check now the mechanism of ischemia in chronic syndromes, and we talk about the microvascular coronary arteries, of course. We have two mechanisms. The first one refers to the structural deficiencies. First of all, we may have inward arterial remodeling, we may have capillary air function, intravascular plugging, perivascular fibrosis or infiltration, and sometimes we may have extramural compression due to hypertrophy or increased left ventricle and diastolic pressure. We may also have a functional deformities. We have impaired vasodilation, which comes due to endothelial dysfunction or autonomic dysregulation, and which then results to increased vasoconstriction in the small coronary vessels. If we review all the studies concerning the epidemiology and the prognosis of this disease, do you know that every patient who has microvascular dysfunction or microvascular angina has a 7.7 more possibility to express a major coronary event, either stroke, either infarction, the past years. So we can see that it's not so innocent, this disease. And it's true. It's true, when we talk about the risk factors, they are exactly the same with the obstructive coronary artery disease. We may have glucose intolerance, hypertension, obesity, smoking. The traditional risk factors remain the same and can lead to microvascular dysfunction. And since I see here too many students, it's very difficult to approach this patient. First of all, we have to evaluate the symptoms, to evaluate whether this angina that our patient refers to us. History taking and physical examination are the first tools we have in order to investigate microvascular angina, and in the second thought, we can perform ECG or even echocardiography. Actually, it's the invasive coronariography who can lead us to our diagnosis. First of all, we can perform in the cath lab an adenosine test and check the FFR, the CFR, and the IMR. But we should also perform a vasoreactivity test, which refers to acetylcholine. And I present you here the protocol. That's the only way to investigate microvascular angina in the cath lab, and how we use adenosine and acetylcholine in order to investigate this patient. However, we have many times that microvascular disease collaborates and coexists with arterial hypertension. And we know about hypertension that we should offer antihypertensive treatment in all patients with coronary artery disease with class of evidence 1A. And it's also true that the first drugs we can use is angiotensin converting enzyme inhibitors, angiotensin receptor blockers, beta blockers, or even calcium channel blockers. And when we have a threshold in order to start the antihypertensive treatment, this seems to be 140 when we're talking about office blood pressure. Also, with class evidence 1A, we know that all patients with coronary artery disease suffering from hypertension, then we should target the blood pressure up for about systolic 130, diastolic about 80. And we know also that the same treatment targets in the general hypertensive application apply also to the coronary patients. From the latest guidelines for chronic coronary syndromes, we know that the recommended blood pressure is 120 till 130, and when we talk for older patients, we can be more flex here and put our targets 140 millimeters of mercury. How we manage this patient with microvascular dysfunction? We have the first thing we have to do is to deal with the traditional risk factors. First of all, dietary changes. We should restrict the salt our patient receives. Physical activity. We know that 150 minutes per week of moderate intention physical activity is the best way to adapt our cardiac muscles, and in some cases we can also increase to 300 minutes per week. Loss of weight, we know that weight management decreases the possibility to express a stroke or myocardial infarction. Smoking cessation, of course, and based on the latest guidelines with class of evidence 2A, we should also cope with stress, as stress is an independent risk factor for manifesting microvascular angina. If you see here an update, the latest years, we see that the first thing we have, as we also mentioned, is the risk factor control. And then we know that the first three drugs we should consider is beta blockers, calcium channel blockers, and nitrates. There's a thing about this, and if we don't see a solution in this, we can proceed to ranolazine, ACE inhibitors, ivabradine, in case we talk with patients with heart failure, or even nicorandil. And when we try to check the latest guidelines, we see that these guides kind of change. So if we put here in this diamond, you see at the peak we have the beta blockers. Right left to it, we have the calcium channel blockers, the hydropyridine, of course, and in some cases we may talk about ranolazine. And it's true that patients with hypertension, especially those who present with left ventricular hypertrophy, it's usually associated with myocardial ischemia and non-obstructive coronary artery disease, which may lead to episodes with MINOCA. And here, with class of evidence 2, we know that the first treatment seems to be RAS inhibitors, beta blockers, and calcium channel blocker. And actually, so every time we have a patient who is hypertensive and suffers from non-obstructive coronary artery disease, our therapeutic scheme should start with assays with a combination of beta blocker, and if he presents with angina, we may add on his therapeutic scheme calcium channel blocker. If he has no angina, diuretic can also be an option for us. And actually, this is a big study, the MINOCA BAT, which took these patients who had the coronary artery disease but no obstruction, and tried to combine beta blockers with RAS inhibitors, check the beta blockers alone, check the RAS inhibitors alone, or received no treatment. And we actually see that the patients who received combined therapy with ASA or ARB plus a beta blocker showed better results concerning the primary endpoint, which seems to be death, hospitalization from MI or ischemic stroke. And when we have the big deal here is, do we treat our patients with antiplatelet drug? Should we give aspirin to patients who have no obstruction in their coronary arteries? We don't know yet. The only study I managed to find is called Warrior. It's in Australia, and it took patients with non-obstructive coronary artery disease, and it's still running. We're going to expect the next 2 years the results, and they add aspirin as an antiplatelet therapy in patients with non-obstructive coronary artery disease compared to those who received none. All of the patients received also lipid-lowering therapy, atorvastatin, and some RAS blockers. Till now, we know that the category which received the intensive medical therapy strategy against those who received no therapy shows better results, except some cases where we have bleeding. As a take-home message, what we should know is that there is a lack of evidence concerning microvascular angina. It's only the past years that we start dealing with this disease. You should know that the blood pressure targets in patients with coronary artery disease seems to be for the systolic one 120 to 30, and for the diastolic one 80. We should start anti-anginal and antihypertensive treatment where the systolic blood pressure is more than 130 millimeters of mercury. The use of assays, ARBs, beta blockers, and calcium channel blockers should be our first therapeutic option, and we should also evaluate the symptoms of the patient. Thank you very much.
Thank you, Dr. Papandianou. Any questions from the audience? If not, thank you very much once again, and we'll continue with the next session.
Our next session, satellite session by Cyprus, Greece, Invasive Cardiology and Cardiothoracic Surgery Working Teams. It's coordinated by Professor Dr. Georgios Georgiou and Professor Konstantinos Lamproulis. We would now like to call upon the stage the chairs, Dr. Panos Huntis and Dr. Michalis Demosthenes.
Good morning, everybody. We'll continue now with the speech of presentation by Dr. Ioannis Zanavaros, with the title is Innovations in Valvular Surgery. Dr. Zanavaros is Director of the Cardiac Innovation Center at Apollonio Hospital and a clinical professor at the University of Nicosia Medical School. Dr. Zanavaros holds a medical degree from the National Kapodistrian University of Athens Medical School. He is a board-certified cardiac surgeon and congenital and pediatric cardiac surgeon. His doctorate thesis was completed at Charité University Medicine, Berlin, where he graduated with magna cum laude. He started his professional journey with a leadership at the Sana Heart Center campus in Germany, where he continued as a resident in cardiac surgery and later obtained his specialty as a cardiac surgeon in 2007. He advanced to roles such as attending surgeon and senior consultant at Sana Heart Surgery, Stuttgart, eventually heading the Department for Surgery of Congenital Heart Defects. Dr. Zanavaros, it's a pleasure for us to be here today.
Thank you for the very kind introduction. I would like to thank the organizing committee for the invitation, and especially Professor Patrikios. I have a quite fascinating topic that has to do with innovations in valvular surgery. I don't have any disclosures. So we have to do with the heart. The heart is a very fascinating pump. The heart begins beating 4 weeks after conception, and that's more than 3 billion beats in a lifetime. You see the heart of the 3-kilogram child beating on the right side. When the heart strokes, it pumps about 70 milliliters of blood, and that's more than 450 million liters in a lifetime. And every day that the heart beats creates enough energy to drive a truck 20 miles. In a lifetime, that's equivalent to driving to the moon and back. Cardiovascular diseases is a leading cause of death globally. Estimated 18 million of people died from cardiovascular diseases in 2019, representing 32% of all global deaths. It is also the leading cause of death in Cyprus in 2020, causing 28% of the deaths. Almost 40,000 hospitalizations in Cyprus are caused from cardiovascular diseases, and also 2,000 deaths every year. Valvular heart disease is any cardiovascular disease process involving one or more of the 4 valves of the heart. Those conditions occur largely as a consequence of aging, but it can also be caused from congenital abnormalities. They are quite frequent, and the prevalence in the States is estimated of about 2.5% in the global population. The frequency increased considerably with age, from 0.3% for the young people, and increasing to over 10% of those aged 75 years and older. Well, still CABG bypass operation is the most common cardiac operation performed, but as you can see, valvular disease operations are increasing rapidly. Of course, the main cause for this is the rapid evolution of transcatheter techniques, which at the time in Germany, they make more than 50% of all the operations performed for valvular disease. That's very fascinating. People were trying to cure heart valve diseases since the beginning of the 20th century. At 1912, Elliot Cutler and Theodore Troufier, and a French surgeon, they tried to dilate an aortic and a mitral valve using this small knife in the dip of their fingers. This was, of course, not very successful, having a mortality of 90%, so those techniques were not established. But 20 years later, at the mid-20th century, 3 surgeons independently found a way to operate heart valves, but those operations were of high risk and of lower efficiency. It was not until the early 60s that 2 surgeons invented valves that could last over time, and was the beginning of heart valve surgery. Years of trial and error were necessary before considerably durable, reliable valves would become available. And I'm asking, what is innovation? Innovation is a new idea or method, or the use of new ideas and methods. Whereas surgical innovations are mostly new and innovative techniques in the practice of surgery and the improvement in the delivery and quality of surgical care. Surgical innovations comprise new techniques, modified strategy, or innovative instruments. For years, median sternotomy was the way to operate on the heart. You have a perfect access to the heart, to the lungs, and to all mediastinal structures. However, nowadays, less invasive techniques like a minimal right anterior thoracotomy incision, where you see on your left, or partial sternotomy incisions are established, and they are very frequently used in order to treat aortic valve stenosis, which is the most common valve disease. Surgeons use modern biological or mechanical valves. They have very low risk for pacemaker. Those valves have lower gradients. Patients recover very fast, and they are quite, they are early back to work after 3 or 4 weeks. An area operation may not be needed. You see the cosmetic results on the patients, which are very happy with these modifications. You can also use robotic-assisted surgery to replace or repair a valve. Those techniques are at the moment not established, and there are only a small case series of patients undergoing robotic-assisted surgery. But this can be a part of our future. Minimal invasive mitral valve surgery is established. Most of the clinics in Germany and all around the world that operate on a mitral valve use minimal invasive techniques. Why this? Because it reduces the surgical trauma, it leads to shorter hospitalization of the patient and quick return to everyday life. It minimizes the post-operative pain and stress, and it's actually at the moment the gold standard of treatment. If the patient has a repaired mitral valve, then he has less thrombotic complications, less hemolysis, a reduced risk of endocarditis, and increased survival rate. How do we repair the valve? Mostly with a ring that's implanted in the annulus of the valve and with a replacement of the chordae tendon. The same techniques can be used to repair a diseased aortic valve using the so-called David procedure, which leads also to excellent long-term results. Our team published also new techniques to replace the pulmonary valve. Here you see one of our patients. She was operated on fallotetralogy. She needed pulmonary valve replacement, and through this minimal incision, we managed to replace the pulmonary valve, and our results were published in the Journal of Surgical Case Reports. Surgeons have now innovative valves. Here you see the results, the 7-year results of the resilient tissue valve, which has a 99% freedom from structural deterioration after 7 years. It is a benchmark for future surgical or transcatheter prostheses. Speaking about innovative valves, I have to mention the decellularized homographs. Decellularized homographs show less hematogenicity and therefore hardly any calcifications. On the right side, you see the first implantation of a tissue engineering valve, a decellularized homograph in Cyprus, performed in a very young patient in our clinic. As I said, the cellularized valves have excellent results because of the reduction of the hematogenicity, and results from Hanover showed a freedom from reoperation after implantation of such a valve of 100% after 10 years. Why are those minimal invasive techniques possible? That's possible because a lot has changed in our everyday practice. We have innovative machines and innovative instruments to assist us for those operations. Technologies like 3D echocardiography and CD imaging offer detailed visualization of cardiac structures. Modern heart-lung machines have evolved to be more compact, safe, and efficient, and minimal invasive tools help us perform endoscopic procedure with very high precision. I have to mention also the innovative strategies post-cardiac surgery. You might not know that 80% of the patients having morbidity after surgery, this 80% occurs outside of the operation room. So we don't only have to fix our operation techniques, but we have to help patients recover faster after surgery. So in our clinic, we use the ERAS strategy, the enhanced recovery after surgery, which is a multimodal perioperative care pathway designed to achieve early recovery for patients undergoing major surgery. The two elderly patients on the right side had, the lady had a 3-valve surgery, and the sir on the left side had a 2-valve surgery and ascending aortic replacement. They were extubated very early after surgery, and they were enjoying their breakfast just the next day in the morning. So, to conclude, heart valve disease represents a major burden for the modern society, with the frequency increasing to over 10% to the patients over 75 years and older. Surgery to repair or replace a stenosis or regurgitant valve is rapidly evolving, and innovative surgical techniques, valves, machines, and instruments have significantly advanced cardiac surgery, enhancing precision, safety, and outcomes. Thank you for your attention.
Okay. there any question from the audience? Okay, yes, sir.
That's an excellent question, and it's a breakthrough. The patients post-cardiac surgery, especially after major cardiac surgery, were kept intubated for a day just to see how they develop. This leads to pulmonary infections. This causes other problems. They need more medication, and we have changed radically this concept. Patients are extubated either in the operation table or directly after in the intensive care unit, and they are mobilized the day after. They have to walk very early, and all catheters are removed very fast. This is good because we reduce the infections. This is good because we mobilize the patients earlier, so they recover faster and they return back to the daily activities very early. This is a major breakthrough.
Okay.
Thank you.
Excuse me, I think Dr. Georgiou would like to ask a question. Excuse me, one question from Dr. Georgiou.
There is one more question. Okay, there is one more question. Yes.
George, wait for the microphone because we cannot hear you. Thank you, Janos, for coming up back up.
Just the question that address you before. So if you do so well extubation, for example, you say, okay, I understand in the ICU, but early when you finish the operation on the table, and if you have any bleeding, for example, what do you do with these patients? Because we all know that postoperative bleeding is one of the major risks of cardiac surgery.
Well,
you reintubate the patient. You don't jeopardize. My question is, you don't jeopardize by this doing the extubation on the table. You don't jeopardize the safety of the patient.
And not at all, not at all. You know, we have to change our mentality to go to have progress. I understand what you mean, but you need meticulous hemostasis in the operation, and you have to be sure that that everything is okay. If you have a feeling that something is not good, you don't have to extubate him. But believe me, this is less than 5% of the patients. We really need to change our mindset. And we operated on 1,010 patients. More than 95% were extubated within the first 1 or 2 hours after the operation, and only 4 needed reoperation due to bleeding, and you have to intubate them again and put them in the operation table. That's not a reason to keep somebody intubated.
Of course, I'm not saying about these patients you say 1, 2 hours or 3 hours after surgery.
Yeah.
I'm talking about those patients which are extubated immediately after surgery. We all of us who do this, we know that the patients they have excruciating pain. A lot of times, I mean, when you extubate them too early, I'm not talking in ICU, in care, I'm talking about the table. And also, all of us, none of us wants to have a bleeding. This is true. No. But sometimes it happens. Maybe it's not because of surgical bleeding. Maybe sometimes it's because of the medications or the coagulations the patients were taking before.
I tell you again, the problem is our mindset. We have to think a little bit different, and there are medications now that help us extubate the patient and keep him calm, like Dextor. we use Dexter, it's quite expensive, but that calms down the patient. The patient is very relaxed. He's extubated, he's breathing on his own, but he's not on stress. The blood pressure does not rise. So there are ways, there are medical ways. That's the development also from anesthesiology that helps the patient calm down, be cool. He doesn't have to have pain, and all together help cardiac surgery evolve.
Okay, well done.
Thank you.
And just a comment, I don't know if you agree. Okay, cardiac surgery evolved through the years, I mean, the industry, for sure. But just, I want to make clear that, because there are many cardiologists around our audience, apart from the people here, minimal cardiac surgery, it's not the same like the general surgeons. They are saying, for example, they do a laparoscopic cholecystectomy. It's actually minimal access cardiac surgery. I want this to make clear, because I see also here in Cyprus, a lot of cardiologists, they have, I think, this misleading, which is actually minimal access, because the patient, again, is passing through the the bypass machine, or the inflammatory problems that, issues that may arise on this. So I think it's just to make clear
That's
the cardiologist, I mean, what we mean when we say minimal invasive cardiac surgery.
You're absolutely correct. Maybe the only example of minimal invasive in a maximal way is off-pump CABG. You don't use the pump. You're operating on beating heart, but the incision is big. This is correct.
Okay, thank you very much.
We can welcome our next speaker. It's Dr. Sotiris Moraitis. Dr. Moraitis is a consultant cardiothoracic surgeon and the director of the commanding officer of Naval and Veterans Hospital in Athens. He graduated from the Athens Aristotle University Medical School in Thessaloniki. He was trained on cardiothoracic surgery at the Evangelismos General Hospital of Athens and at the Harefield Medical Hospital in Middlesex. He holds an MSc degree on thoracic oncology from the Medical School of the National Kapodistrian University of Athens and an MSc degree on endovascular techniques from the Medical School of the National and Kapodistrian University of Athens and the Department of Surgical Sciences at the University of Milano Bicocca Monza, Italy. An MBA degree on healthcare management from the Department of Economic and Political Sciences at the National Kapodistrian University of Athens and a PhD from the Medical School of the National Kapodistrian University of Athens. He's going to talk us about the transcatheter aortic valve implantation, known as TAVI, versus the sutureless aortic valve replacement. Dr. Moraitis.
Mr. Chairman, thank you for your kind invitation. First of all, I would like to thank the organizing committee and especially Professor Ioannis Patrikios for his kind invitation. Yanni, it's a pleasure and an immense honor for me to be among you. These are my disclosures. As we all know, TAVI is a minimal invasive procedure that inserts a bioprosthetic valve VA catheter through usually the femoral artery, but has shown excellent short-term outcomes and some concerns about long-term valve durability and potential valvular leak. This is the first performed TAVI by Alain Cribier in 2002. And this is the development of TAVI. Allow me please to use the term TAVI because there is no replacement, there is just an implantation. The native leaflets are still there, the calcification is still there. On the other hand, sutureless SAVR uses a bioprosthetic valve that can be implanted without traditional sutures, significantly reducing operative times and myocardial ischemia duration, and it combines the durability of surgical implanted valves with a reduced recovery period of minimal invasive techniques because you use mini thoracotomy or mini J sternotomy or mini sternotomy. This is the first sutureless valve in the market, is the Perceval. We have to say that you may notice the high cannulation point and that we have to make an aortotomy at least 5 centimeters above the sinotubular junction. These are the sizes, and this is how the valve, sorry about that. This is how the valve looks. The sizer, the transparent sizer, has to go through the annulus and the white one not. And of course, you can see on the bottom line the correct sizing. We use 3 guiding sutures placed at the 2 or 3 millimeters above the commissus and through the eyelets of the valve. The eyelets are here and here. So we use the guiding sutures to guide the valve through the delivery system, and then on 12 you may see how the valve is deployed, and this is how is the correct sizing or the oversizing. The other valve is Intuity by Edwards. It's a rapid deployment valve. It's not exactly a sutureless valve because sutures are stayed in place, and you may see here that there is a skirt of the valve which is opened in the outflow area of the left ventricle. Why do we need these valves? Because on 2023, this paper published saying that all surgical supraannular aortic valvular tissue prostheses are labeled too large. So you may see here the 8 valves that they are in the market labeled as 27 millimeters, and the real diameter is from 21.9 to 25.8. That gave us the opportunity to read on 2024 the state-of-the-art review in the JAC, talking about the wireless option for the sutureless valves, highlighting that these are the 2 valves, the Perceval and the Intuity, and saying that potential candidates for SUSAVR include those undergoing concomitant procedures, complex reoperation in patients with frail aortic root tissue, minimally invasive aortic valve replacement procedures, and of course, high-risk patients for whom TAV is not suitable. These are the characteristics. You may notice that the one valve is delivered collapsed. There are no sutures. There is a high cannulation and a high aortotomy instead of the other one. And here we cannot miss that there is a reference about the cost, which is extremely important. And you may see here unavoidably that the cost for TAVI has been increased to 80%. So this is the algorithm. We don't have to go through it right now, but just keep in mind that SUSAVR is a great alternative of TAVI, especially for the patients that they have the characteristics in the blue shadow. The comparison revealed that the mortality, the 30-day mortality, is reduced with SUSAVR, while the return to daily activities is quicker with TAVR. As far as concerns for hemodynamics, the results were almost the same. But many papers came out the last 2 years about the sutureless and the transcatheter valves, concluding this paper of 2023 that TAVI patients had a significantly lower 5-year survival and survival freedom from major ACCEs with a higher rate of PPI, pacemaker insertion and paravalvular leaks when compared with the SUSAVRs. The blue line is the SUSAVR and the red line is the TAVI. Another enthusiastic paper saying that SUSAVR is more advantageous compared to TAVI and can be considered a valuable and promising option for the gray zone of aortic pathology, which is that gray zone. The gray zone patients may have moderate surgical risk because of the comorbidities that increase the risk of conventional surgery, but do not necessarily make them high risk enough for undergo a TAVI. And I think the truth is exactly presented by this paper here. That's the main difference. The transcatheter valve is deployed under rapid or controlled pacing, making fine adjustments in depth and angle challenging due to calcified leaflets and blood flow effects. That's why it's TAVI and not TAVR. In contrast, SUSAVR is performed under cardioplegic arrest, allowing for precise placement with guide sutures, which theoretically limits deeper implantation. This is a multi-institutional European study of last year under Professor Claudio Muneretto, and this is the graphical abstract. You may see here, you may notice that the outcomes are better than the TAVI. And a part of this study, where one of today's keynote speaker, Professor Di Bartolomeo, was participated, suggesting that the extension of TAVI to low and intermediate risk patients should be evaluated with caution. The blue line is still the sutureless valve, and the red line is still the TAVI. This is another paper from the University of Louvain. The results are almost the same with the previous one, and University of Louvain is one of the centers that they use the Perceval valve for a long time, since 13 years ago. This paper suggests that the take-home message should be that the Perceval valve offers a safe and effective treatment for aortic valve disease, minimal invasive single valve and combined cases. Another enthusiastic paper of this year saying that sutural valves bridge the gap between conventional and transcatheter aortic valve replacement. These are some of our cases. You may notice the frame of the valve, the high aortotomy, and how the frame is attached to the aortic wall. This is how it looks on the CT, and of course, because not everything is going well, you may see here one of our cases after distortion of the valve 1 month after the implantation, causing a paravalvular leak. And when you compare the hemodynamics and the clinical outcomes, unavoidably, you compare the cost issue as well. So TAVI is expensive. And having said that, there was a paper on 2022 suggesting that patients who undergo surgery after heart team evaluation show larger lasting results and more favorable cost ratio if they had the SUSAVR. Again, to the University of Louvain, Marie Laberitz, in the last act, said that the mean cost of SUSAVR is 14,000 euros compared to TAVI, which is 24,000 euros. If we try to find cost-utility studies, we'll see that SUSAVR are expected to live an average of 1.25 years more. And of course, the cost-effectiveness studies revealed that the SUSAVR method provides an additional 1.14 quality-adjusted life years compared to TAVI method. And having said that about cost, we have to discuss what's going on after the transcatheter aortic valve replacement, implantation, with the cardiac surgery. So this is a paper of this year. SAVR after TAVI is the fastest growing adult cardiac operation in the United States. Why is that? Because practice does not follow guidelines. This is an article in press saying that in the state of California, were identified 2,360 patients under 60 years old And that means that this patient had to undergo a mechanical valve replacement. The conclusion is that despite consensus guidelines, bioprosthetic shower and TAVR, why we do bioprosthetic shower, because if there is a need of a reoperation, we can put a TAVI. So bioprosthetic shower and TAVR volumes are nearly equal in patients under the age of 60. And in proportion-matched patients, TAVR is associated with inferior 5-year survival, increasing the need for randomized trials to inform clinical practice. To conclude, innovation in prosthetic valve processing are expected and hoped to improve clinical outcomes. The use of SUSAV appears to offer a higher value of QALYs at a lower cost than TAVI, but the effect is not statistically significant yet. Definitely, the patient benefit outweighs the cost, but no one knows if expensive treatments pose at risk to the sustainability of health system budgets. We had a great discussion with Alain Cribier in September of 2023 about the cost of TAVI. Unfortunately, Alain Cribier died last February, on the 16th of February. He will be widely missed, but his legacy will go on and on as long as people have aortic valves which get sick. Thank you very much for your attention.
Thank you, Dr. Moraitis. That was an excellent talk. I only have one question. How do you treat paravalvular leak on a sutureless valve discovered 1 month after the operation? Is ballooning enough, or do you have to go? Yep.
Usually, balloon aortoplasty is enough, usually. The second alternative is to use a TAVI.
Okay.
But definitely, we can avoid the reoperation. Even that there is a certain method we can remove the sutureless valve without destroying the aortic root. But definitely, balloon aortoplasty will be the first option.
Okay, thank you. Any questions from the audience? Dr. Georgiou?
You very nice mentioned also the economic impact of TAVI in now existence. But I personally think that our main resource that we have as cardiac surgeons against TAVI, it's the well-known bioprosthetic valves, which they have a longevity, and they have been the evidence we have for the last 20 years at least. And the cons which goes with the TAVI is that we really don't know, despite that we see it's biological valve, we really don't know the life expectancy of it. But to be honest with you, I believe that TAVI, okay, is here and will stay. It's just, as you correctly taught in the US, that anyone does what he or she wants, which this is not correct. And we are lucky that, like you have a committee in Greece, we also have now a committee here in Cyprus, which has to get this permission to do TAVI procedure. I mean, the real heart team to be there, not just to say, wait, okay, heart team. The interventional cardiologist asked the surgeon told him, okay, it's difficult, proceed. I mean, this patient is difficult, proceed with TAVI.
Thank you very much for your comment. I absolutely agree with you. Definitely, the guidelines are there, and we have to keep them. The thing is that TAVI, as you said, came and is here to stay, but not for everybody. And definitely, there are many patients that went very well after TAVI, but the thing is that, let's say the companies are creating needs more and more. The thing for the public sector, the health public sector, is that there is a great issue with the health economics, and TAVI is against the health economics we need in the public health sector. And as a part of servants in the public health sector, I have to point out that and highlight it.
Thank you very much, Dr. Moraitis.
Unfortunately, due to the personal circumstances, one or two of our presenters won't be able to come today. Therefore, please join us for this brief coffee break at the back of the conference room, and we will resume in 15 minutes. So, see you. Ladies and gentlemen, please be seated. We're starting soon. The next session will be Interventional Cardiology Advancements, coordinated by Dr. Vlasis Ninos, Dr. Athanis Konstantinos Toutouzas, and Dr. Michalis Demothinos. We would now like to call chairs Dr. Savas Konstantinidis and Dr. Konstantinos Diplaris.
Okay, good morning, ladies and gentlemen. It is a pleasure to chair this very interesting session on interventional treatment of valvular heart disease. The first speaker is Dr. Konstantinos Parias, who's going to talk about transcatheter tricuspid interventions. Dr. Parias needs no introductions. He's a UK and Canada trained cardiologist specialized in interventional cardiology and in particular valvular interventions. He completed his PhD thesis in the University of Leeds while in training there and his clinical interventional fellowship in Vancouver, trained and mentored by pioneers such as Dr. Webb and Dr. Penn. He returned to Greece to work as interventional cardiologist at Onassis Heart Center, where after some extensive coronary work, he became an early adopter of the emerging valvular interventions. He later moved to IEA Hospital, where he organized and directs the first of its kind department of transcatheter heart valves to date. Kostas, we are eager to hear you.
Hello, so thanks for the kind introduction. I hope you can hear me well. You can see my slides, right?
Yes, we can. Thank you.
Perfect. So, hello, good morning from the exotic Lemesso, and thank you for the organizers and Professor Patricios for the kind invitation. So my topic today will be the current tricuspid valve therapies and the landscape we have today and what we expect for tomorrow. We all know all the clinical signs and symptoms of tricuspid regurgitation that are associated with low cardiac output, and the symptoms like fatigue and low exercise tolerance, of elevated central venous pressures like edema, anasarca, ascites, and the commonly coexisting atrial fibrillation. And the etiology of the tricuspid regurgitation is mainly secondary, mainly is an indicator of left ventricle disease, either valvular disease or muscular disease, and very rarely is primary, like rheumatic disease, myxomatous disease. And so as a secondary etiology, as you would expect, this is mostly a marker of the underlying primary etiology. And the symptoms are very unspecific. There's a long latency period, and the only medical treatment, drug treatment that alleviates symptoms are diuretic. It's certainly an indicator and prognosticator, and even as you can see, moderate tricuspid regurgitation is independently associated with long-term prognosis. It's a very frequent disease, as you can see here from the US survey. It's the second most frequent valvular disease, second to mitral regurgitation. So a lot of people are suffering from tricuspid regurgitation, as you can see in the small blue boxes. Proportionally is the least treated mitral valve disease. And the main reason for this might have been the very unfavorable surgical results. So when we operate on isolated tricuspid regurgitation, as you can see, the operated mortality is almost 10%, and there is no trend of reduction over the past year. So there was an unmet need for a low-risk transcatheter therapy, clearly in the tricuspid valve, and here we are. And presently we have these commercially available transcatheter therapies. One is with the clips, and the other is with a transcatheter valve. So the clips we're using today is the TriClip and the Pascal, a very similar mechanism of action, just a different design and a different way of closure. As you can see here, a TriClip is opening and closing very firmly, like a vice grip, whereas the Pascal, it has a nitinol frame, so there is an elasticity there, and it can accommodate some movement. And here you can see the two cartoons of these two clips in action. And so you can see as we advance from the femoral vein, we're just in front or to the back of the tricuspid valve. And then we are targeting the coaptation zone where the tricuspid regurgitation is originating from. And we bring close together the two leaflets there, and we capture them, and then we close the device. And this is the treatment, so simple. Sometimes we have to position the second clip to have a better outcomes, as you can see here. So this is a typical result we're getting. You see this torrential tricuspid regurgitation coming from everywhere. You see we have positioned two Pascal ACE devices, and this is the final result. Quite typical, almost no tricuspid regurgitation anymore, as you can see also from the 3D color and from the glass view. The TriClip comes in 4 different sizes in terms of length and width, and it's supported from the TriLumina trial. We've recently had a TCT, the 1-year outcome. So this was a randomized trial to a TriClip versus optimal medical therapy, almost 600 patients included. And you can see here how the tricuspid regurgitation severity was reduced in the TriClip arm, but remained the same unaltered in the optimal medical therapy group. So if you notice here, the green colors, 50% of the patients at 1 year had mild or non-tricuspid regurgitation. This is the number we should targeting for, because the other 38% had moderate tricuspid regurgitation. This was a positive study in terms of the primary endpoint, which was a combination of clinical outcomes and clinical improvement, significant clinical improvement, as assessed by questionnaires like the KCCQ score, when it was an increase of more than 15 points. In terms of mortality, as you can see, there was no difference at 1 year between the group. In terms of heart failure hospitalization, numerically, there were less in the device group. But what was really significant was the symptomatic improvement as assessed by the KCCQ score. And what is important mechanistically is to see that the more you reduce the tricuspid regurgitation with this therapy, the more the clinical improvement you see. A similar study also reported this year was the TriFrance, a similar randomized between TriClip and optimal medical therapy. Similar primary endpoint, also positive study, better doing the patients at 1 year, and the reduction of the tricuspid regurgitation similar. So the mild to non 33.5%, and the rest 37% was moderate reduction. Now just showing this to show you that this is a very benign procedure. The in-hospital death was below 1%, which is an excellent result. And on average, 2 clips were used per case. This is the 2nd generation Pascal we're using today. It comes in 2 different sizes, the classical and the smaller one. It has a different design, as you can see here. I'm not going into details. And the TriClasp study was a study that we participated also from a year with 300 patients enrolled. It was like a single-arm study and a follow-up study. And here you see how the admissions for heart failure were reduced in the year post the procedure compared to the year before the procedure. And here is the reduction of the TR grade, as you can see, approximately 60%, the very good result, 0 or mild tricuspid regurgitation at 1 year. So excellent result in real clinical practice. This is another registry with Pascal, the PACE registry coming from large volume centers, similar results at 1 year. As you can see, 40%, a very good result, 80% if you also include the regurgitation, so the moderate regurgitation. So where the clips were positioned in this registry, as you can see, the site of the clipping was mainly the anteroseptal commissure, septal commissure, anterior, posterior, and less the posteroseptal commissure. And what we noticed in this registry is that more doesn't necessarily mean better. Actually, as you can see, the more devices they were positioned, the less successful was the procedure. So when you have to position many devices, it means you're not very successful or you are tackling very difficult anatomy. And of course, again, when the coaptation gap in the center is large, then you are less successful. What we are all waiting very eagerly is the Class 2 TR study, which has been completed enrollment and will be reported sometime next year. The largest study so far, randomization between Pascal and optimal medical therapy. So in summary, when we are dealing with clipping of the tricuspid valve, our real success is somewhere between 35 and 50%, which is not that great. And why is that? This is because we have very difficult anatomies in the tricuspid valve. We are clipping three leaflets and not 2, like in the mitral valve. And moderate tricuspid regurgitation is associated with mortality. So we cannot just skip it and say that's okay. So we should have a score to predict when we're going to be unsuccessful. And then we take these patients, which is almost 30% of the patients we're doing today, that we're not getting great results. And also all the patients we reject for clipping from the outset, which is approximately 30% of the patients we screen. So a total of approximately half of the patients we see for transcatheter therapy of the tricuspid valve are not good, are not perfect for clipping. And these are exactly the patients that we should be looking for another solution. And this another solution is now available from the beginning of this year, and this is transcatheter heart valve with the EVOQUE valve. This is the EVOQUE valve. It's a nitinol frame valve with 9 hooks. It now comes in 4 sizes. We also have available, and we started using the 56 millimeter EVOQUE. And this is just a cartoon of the procedure. You go with the capsule of the valve into the tricuspid valve, and then you position it coaxially in the center, and you gradually unsheath the valve. First, the hooks are exposed, and they capture the leaflets. And then with slight manipulations, you release the main frame of the valve, and once you release it, it starts working. Is it successful, this therapy? Of course, because the CE mark and FDA approved, and this is the study that supported the EVOQUE valve, the TRISCEND 2. It was also reported at TCT a few weeks ago, the 1-year outcome, randomized study, as you can see, more than 400 patients to EVOQUE versus optimal medical therapy. And here, we don't have all the questions, are we going to be successful or not? We're almost always successful. You see the TR reduction, the good one, is more than 95% of the patients. So you position the valve and the tricuspid regurgitation is gone. And then the results were pretty decent at 1 year. You see the mortality at 1 year was less than 6% and lower than the mortality in the control group. We just noticed the need for the pacemakers, which was higher in the EVOQUE group. This was a positive study. It was a combination of the primary endpoint of clinical outcomes and improvements in symptoms, significant improvements in symptoms, like all the previous study, and it was a strongly positive study. Numerically, there were trends for better outcomes in terms of mortality and in terms of hospitalizations, unlike the TRILUMINATE trial. And there was significant symptomatic improvement with 18 points of KCCQ, a questionnaire that is a real big symptomatic improvement. So These improvements mean that if you treat 5 patients with EVOC, then you have 1 that is alive with very large improvement. If you treat 3, then you have 1 which is alive and is feeling well. Are we done? No, we're not done, because we're not going to see further improvements in the clipping field with clips that will be elongated, expanded, transformed, and be more successful. And we're also going to see more transcatheter valves coming soon. And I'm going to finish my presentation by just demonstrating an EVOC implantation. You see this tricuspid regurgitation with a huge gap that it will be impossible to grasp and to clasp with a clip. So these are exactly the patients that we know clipping is not going to be successful, and we had no treatment option before EVOC. So here is the EVOC, and you see the final stage of the release of the valve. So this is how you're going to see the valve in the future in the chest x-rays in the wards. And this was the result, an excellent result with no paravalvular leak or transvalvular leak, as you can see in the 3D zoom with color and in other 3D views. And if we have the time, I'm going to show you another case very quickly, a complex case with previous Pascals in the mitral valve for mitral regurgitation and an electrode of defibrillator. You see how thick it is. This is not a problem for EVOC. We do our screening, we decide the valve size, and we believe that the electrode was responsible, partially responsible for this huge tricuspid regurgitation. As you can see, the electrode is hitting the septal leaflet, and then we are coming with the EVOC valve, and this is the positioning initially of the wire, then the capsule, all the monitoring is with echo, and gradually exposing the valve, and we check at every stage that the leaflets are inside the hooks, and gradually continue to deploy the valve and keep it coaxial, and finally deploy it, and we get again this excellent result, very typical of EVOC implantation. You eliminate the tricuspid regurgitation, and the valve is where it should be. And with this, I would like to thank you, and I'm here to answer any questions you might have. Thank you for your attention.
Thank you, Kostas, for a fantastic talk. Do we have any questions from the audience? Can I ask you a question, Kostas? We used to refer to the tricuspid valve as the forgotten valve. So obviously there is an issue of patient selection, and when do we decide that the patient is suitable, or is the time to intervene with the tricuspid valve? Could you give us a brief outline of when and how you select your patients?
That's the 1 million question, Salvas. But we now started to understand better when we should intervene, because we have these treatments, transcatheter therapies, for quite a few years. I think the sweet spot point is when the patient starts getting symptoms, but before advancing too much and starts getting symptoms like of right heart failure, ascites, anasarca. So we shouldn't intervene in a patient who is completely asymptomatic, but once the symptoms appear, we improve the patient with diuretics, and then we should proceed to the therapy. Because if we leave the tricuspid regurgitation, as we know, with regurgitation begets regurgitation, so there is dilatation of the right ventricle, and then every therapy is becoming more difficult. And then in terms of therapy selection for transcatheter therapies, we should check the anatomy. And as I pointed out earlier, I think in approximately half of the patients our judgment will be that clipping is not going to be very successful, or the likelihood of success will be low. So in those patients, I think we will proceed with the EVOC valve with transcatheter replacement.
Thank you.
Hello, really interesting talk. Just a quick question, it's more technical on the Evoke system, which is really interesting to, as a concept. Is it possible during deployment to reposition it, or once it's opened and attached to the inner part of the right ventricle, it stays there?
Yeah, that was a typical question of all the implanters when they begin the program. I think, not a thing, this is a reality. Once the hooks are outside of the capsule, then you're bound to continue the deployment. You cannot remove the system because you're going to injure the heart. But you can decide whether you're going to proceed or not before exiting from the capsule. And what is really important in this procedure is to attain coaxiality before starting the deployment. So if you're really coaxial in all axis, anterior, posterior, lateral, septal, and if you can position in the center of the valve with the right depth, then the procedure is essentially finished. Everything else, it's just very typical after that. So yes, you can make this decision and you can remove the system before you exit the hooks from the capsule. After exiting the capsule, you cannot receive or withdraw the system without problems.
Okay, thank you very much. Okay, if there are any other, if there aren't any other questions, sorry, we can proceed to our next talk. It is Dr. Vlasis Ninos, a good friend. He's an interventional cardiologist, currently serving as the director of the cardiology department at the European Inter-Balkan Medical Center in Thessaloniki, Greece. His main interests lie in the interventional therapy of valvular and structural heart disease, as well as the interventional treatment of heart failure. Dr. Ninos has pioneered clinical research in these fields, participating in numerous pivotal trials and significant first-in-human and early feasibility studies. His contributions have been instrumental in advancing interventional cardiology, particularly in the areas of valvular and structural heart disease therapy. He's joining us online, and his talk will be the question I usually ask him when we meet. What transcatheter mitral valve interventions, where are we standing today?
Good morning. Hello, Kostas. Hello, Savas. It is a great pleasure to participate in this amazing Congress. I'm really sorry I cannot be there as a person, physically present, to share a coffee and a drink with you. Some family issues kept me away from Cyprus, which I love very much, as you know. I particularly want to thank Professor Patricios for this invitation. And the topic that I will present is, as you mentioned, is the transcatheter mitral interventions. Following Kostas' talk about the forgotten valve, which is no longer forgotten, we're just going to now focus to the other AV valve, the mitral, which of course we know that there's a huge unmet clinical need, and we have been trying to deal with this valve for many, many years. These are my conflicts. So mitral valve disease is common and is versatile. There are many phenotypes similar to the tricuspid that we just discussed. The surgical options are there. They have been there for many, many years, much more options compared to the tricuspid, especially with the new advances of the endoscopic surgery. We recently had the endoscopic World Endoscopic Meeting in Thessaloniki. However, there are particular phenotypes that surgery has limited value, and that has been recognized for a number of years. So ventricular functional mitral regurgitation and also mitral annular calcification have been notoriously the Achilles heel for surgery, but also for interventional therapy in some aspects. However, the transcatheter therapies have evolved enormously over the last 15 to 20 years and now have ample evidence to support this. Starting with the MitraClip that has now completed 21 years since the first human application in Venezuela, and with a number of publications starting from the first trial that compared it with surgery. in a very bold way that showed superior safety, mainly in the blood transfusions and similar clinical improvements, but inferior performance in terms of the reduction of mitral regurgitation with this initial clip that was used between 2008 and 2011. And going back to the two recent trials, first is the COAP trial that has compared clip versus medical therapy in 2 and 5 mitral clip intervention versus medical therapy in patients with moderate mitral regurgitation. This was a Greek trial. Kostas and his team and our team has put most of these patients in this trial, and there was simultaneous publications in New England and JACC. You can see Greece has contributed 237 of the 505 patients in the cohort. The rest have been contributed by other countries. In an investigator-initiated trial, Stefan Anker was the PI, so it's a heart failure-initiated trial, which is very important. And you can see the median anti-proBNP levels have been 2,600, so somewhat lower compared to COAP trial, which is about 4,500. The median effective regurgitant orifice area has been 0.23. However, the regurgitant volume has been 35, similar to the COAPT. But the volumes of the left ventricle have been a little bit higher compared to COAPT. So COAPT had volumes, and diastolic volumes around 190. This was 200. There was another trial, the MITRA-FR trial, that had diastolic volumes around 250, so bigger ventricles, so that had a neutral result. But ReShape also had a very positive result in the combined endpoint of heart failure hospitalization, cardiovascular death within 24 months. And you can see that the number needed to treat has been about 8. So similar magnitude of benefit compared to COAP trial. And of course, we had improvement in every aspect of quality of life. So KCCQ, summary score, NYHA class improvement, 6-minute walk distance have been massively improvement by the intervention. And because we've put a huge number of patients in Greece, we actually saw the benefit of these patients. So that was a benefit that we saw for the years of the follow-up. So this is the improvement of the KCCQ. So there is a question, maybe, we have discovered a third population, so a population with slightly bigger ventricles to the COAPT and slightly less mitral regurgitation that derived significant benefit from the intervention. But nevertheless, this is the second randomized good quality trial that actually shows a significant benefit to this population. The guidelines as they stand now for the secondary mitral regurgitation still incorporate the COAPT criteria, so ejection fraction between 20 and 50%, and systolic diameter less than 70 millimeters, and pulmonary arterial systolic pressure less than 70 millimeters of mercury. And this is the same criteria for both ends of the Atlantic. So America and Europe share the same 2A indication for mitral intervention. In the same guidelines, the surgery, isolated surgery for functional mitral regurgitation for this population has class 2B. 2A is only when combined with revascularization, so bypass surgery. This is a patient that can actually, we can actually start to talk about lifetime management considerations of MitraClip. So this is a dilated cardiomyopathy patient 11 years later. This patient was in really bad shape even after getting a CRT therapy 11 years ago. He had a MitraClip. He was in and out of the hospital with proBNP going up to 15,000. He couldn't tolerate any medical therapy because of low blood pressure. He got the MitraClip, his situation improved, hemodynamics improved. He started getting better guideline-directed medical therapy. And now 11 years later, he's able to take ARNIs and Tresto as well as SGLT2. So it's very important that this is not an isolated therapy. This is a hybrid therapy that can actually facilitate further improvement of medical therapy in this population. So this patient actually survived in order to see new drugs getting into his system. He even took Vericiquat recently, which is another medication that he's able to tolerate. Of course, MitraClip has evolved. As Kostas mentioned, there are 4 sizes for the mitral and the tricuspid. We normally use the long and wide clip most of the situations for this population. The Expand G4 has demonstrated in real life patients what is the efficacy and the safety of the device in over 1,000 patients, so you can see what to expect. At 1 year, you should expect mild or less, so 1+ or less residual MR in 92.6% of the patients. Depending when you break it out to primary or secondary mitral regurgitation, primary is less than 90%, secondary can be as high as 95%. Of course, this was real-life population, but there was a lot of moderate mitral regurgitation patients. But it's quite interesting to see what is the current efficacy of the therapy compared to coapt. So now we can get with the Gen 4 mitral clip 95% MR +1 or less. With the coapt, we used to get 67%. And this is something that we saw in the ReShape trial because we started in recruiting patients for the ReShape trial with Gen 1 and Gen 2 mitral clip, and our efficacy was reduced. But the latest cohort of the ReShape population was with Gen 4 clip, and we did see a lot of improvement in the outcomes in these patients, the technical and procedural outcomes. And this is what Gen 4 showed. What is the heart failure hospitalization rate for the cohort, 16.9%, and the 1-year mortality, 12.3%. So this was a patient mostly with moderate MR. ReShape was a population with moderate MR. And interestingly, the 1-year mortality in ReShape has been, in both groups, has been around 12%, whereas coapt with more severe MR had a mortality around 23-24%, which is very interesting. What Expand G4 has shown, what ReShape has shown, what coapt has shown is the outstanding safety of the intervention. Complications such as stroke, infarctions, bleeding, detachment, leaflet damage, mortality acutely are very, very low. They are close to zero. 30-day outcomes, 0.2, 0.5. So it's a very, very safe intervention. And of course, Pascal is here for this population too. We have the CLASP 2D randomized comparison between Pascal and the mitral clip for degenerative mitral regurgitations patient. Again, outstanding safety for both devices, and non-inferiority was met, and improvement in the functional class 6-minute walk distance for this population. But TEER is very effective. Why do we need replacement? Because there's a lot of research going on for replacement, because there are anatomies that cannot be treated by TEER. And there is still some residual MR, and we do have recurrence of mitral regurgitation in some group of patients, after 1 or 2 or 3 years. So we're left with a clip in the middle, knowing, having nothing to do with this population. And then there is this philosophical question: why can we not achieve a 0 MR like you saw with the Evoque in the tricuspid? Why shouldn't we have a TAVI-like outcome in this population? And we can do that. I mean, this is bioprosthesis in the mitral space with degeneration after 11 years, severe stenosis. We can put a valve in valve transfemorally, transeptally, without much of a hassle, really, and have an excellent outcome on this population. Or we can put valve in ring even after modification of the anterior leaflet. Sometimes you can see this implantation, and that can actually work very well for these patients. You can see the acute and intermediate outcome for this group, or even valve in MAC, which is a more laborious procedure. This is a novel technique that we started to use with Batman, so balloon dilatation of the anterior leaflet and then implantation of the valve inside the leaflet to get it out of the way from the LVOT. And this is the acute result. So why can we not just do that? Because, of course, mitral valve disease is very complex. It's not aortic valve disease. There are lots of buttons. And, frankly, replacement has been the forbidden fruit for interventional cardiology. We saw now that there is one technology for tricuspid. We have got more coming. Right now, there's no commercial available device for transcatheter, transfemoral, transeptal mitral replacement for anywhere in the world, approved, commercially available. So this forbidden fruit is there, and there have been efforts since 2012. Several devices have been tried. Most of them have failed. Only 2 are still alive, like 3. Intrepid, which used to be 12. Tendyne, which is transapical, of course, and I'm not sure what is the future of transapical therapy. And HighLife was still around with modifications. We made several mistakes. We thought transapical is okay, but it isn't. we put big sheaths from the femoral approach, and that is not compatible with most patients' physiology. Why did we stumble in lots of hurdles? Because there are several anatomical issues there. For example, we have the LVOT situation, the presence of MAC. We have the tier now as a competition to efficacy for this population. And of course, we have clinical factors and size issues that we have to incorporate into our selection algorithms. And the problem is that for most of these patients, at least 65 to 85% of the screening for most of these technologies are screen-failed population. But now we have made some realizations. For example, transfemoral should be the universal approach. It has to be fully percutaneous, not a cut down, minimize damage to the vein. And we have technologies now coming in to incorporate this. All technologies that have improved and new technologies that have new concepts. So, for example, the Intrepid, which is an old friend, and it was tried transapically, now is getting transfemoral, and now 29 French is a reality, and slowly is getting into the trial, the Apollo trial across both ends of the Atlantic. And we have new old friends with new skirts. So, for example, High Life now is Clarity with the open skirt, AltaValve with the supraannular design, and InnoValve that was recently acquired by Edwards with the twist concept of twisting the cords and remodeling the left ventricle. And we have trials. So Summit trial finished with the transapical tendon of the MAC arm is never going to happen for the transfemoral arm. It was a very ambitious randomization effort against TIR, but the MAC arm should be out hopefully soon. Apollo now with the new iteration of 29 French should start enrollment a little bit more actively in Europe and the US. But the good news is that Edwards finished with the Circle trial in the M3, finished recruitment in both cohorts. It's very interesting to see that all the 4 randomized trials had a MAC cohort, so they all want to address the MAC as an important issue, which is very, very important. And AltaValve, which we have experienced in Greece, the supraannular atrial fixated device that sits above the mitral annulus with the annular ring and an annular skirt, finished the EFS very successfully. Up to date, we have 99 patients treated globally. Maybe it's now over 100. And we have seen some very important safety outcomes of the EFS and also quite significant efficacy in eliminating mitral regurgitation and improving patient status in terms of indices of heart failure like KCCQ, 6-minute walk, and also NYHA class, and also some good signals that it can be compatible with MAC. This paper showed 6 patients with severe MAC, and you can see MAC can have many faces. It can be circumferential, it can be at the trigons, it can be thick, it can be thin, but nevertheless, this device seems to be interacting quite favorably with MAC. And this is how it looks. This is a patient that we did as part of the EFS, so transeptal puncture is done in the inferior posterior location. Septostomy is performed using usually an 8 or 10 millimeter balloon. And then the system, which is the steerable guide sheath, 29 French, is introduced through the septum inside the left atrium. You can see this is monitored with echo and fluoro. Is echo-based but also fluoro-based approach. You can see the standard on the table. There is a delivery system now that is introduced through the steerable guide. The system now is flexed down towards the mitral valve. You can see the flex and the positioning above the mitral annulus. And slowly the valve is going to be deployed from the annular ring. You can see how it is deployed, starting from the annular ring there and all the way up to the left atrial wall. And actually the deployment doesn't take longer than 12 to 15 minutes. So it's a TAVI-like deployment, quite fast. You can see how the annular ring is deployed. The valve is fully functional. The hemodynamics are stable. And then the cage is deployed to get apposition against the left atrial wall. Once the cage is opposed against the left atrial wall, the valve is unstable and we can assess the hemodynamics, the echo performance, and then we can deploy and release. And upon release of the valve, we can actually see how it looks on echo. elimination of mitral regurgitation. This is the beauty of mitral replacement. We can assess the gradient through the mitral valve, through the LVOT. You can see the nice anterior tilt of the valve in relation to the LVOT, how it looks on 3D echo. And we can finish the procedure literally within an hour, skin to skin in most patients. You can see how the leaflets are coapting. They're still functional, they're coapting underneath, providing from the ventricular end an active sealing to the technology. And this is how it looks on the transthoracic echo. You can see widely open LVOT and no MR. So in this technology, this is the fourth randomized trial, which is actually running, the Atlas trial. We started recruitment in Greece, and also 2 patients were recruited in the US, so 6 patients so far. It only started about last month, and more is about to come. And there is a MAC arm as well as a primary cohort. So as a conclusion, exciting times are still ahead in transcatheter therapies for mitral regurgitation. MTR is now an established therapy with evidence that expand to a broader patient population. Replacement is on a promising path with many steps remaining to take place, but there is an obvious advantage of commercial available devices that can be deployed transfemorally and fully percutaneous. The elimination of the MR and elimination of the uncertainties in certain patient population of the TEER as an efficacy. But of course, a lot is there, and we're eagerly awaiting to see the results of the trials. Thank you very much for your attention, and I'm very happy to answer 2 questions.
Thank you very much, Vlasis, for the excellent talk and the insight on all the innovation which is already here and the rest that is coming. Are there any questions from the audience? I would like to ask a question. It's regarding the Alta valve that you showed us just now. Does the deployment of the valve impede any future access to the left atrium for either endovascular or open procedures?
Yes, the cage that is made from nitinol, so it's a stent frame which is made from nitinol, which we all know as a material is compatible with implants inside the heart, and there's a very low thrombogenicity and rapid endothelialization. Several devices like the ASD and the PFO devices are made of nitinol, most of them. But what is interesting is that the cells that are there, they are compatible with the passage of catheters up to 24 French. So for example, if you want to close the LAA, left atrial appendage in the future, or perform an ablation of the pulmonary veins, or even put a valve in the future, the access is there, you can do that. And in fact, in a number of patients, we have closed the iatrogenic ASD because there was either right to left shunt or there was a lot of left to right shunt. This is something that can happen more frequently in replacement versus MitraClip therapy because the tubes, the sheaths are a little bit bigger and the holes are bigger. We perform septostomy, but the cage was actually very much compatible with putting an ASD or a PFO device in there. Very, very easy. In fact, easier because you had a fluoro marker on the table, so you could actually adjust the position of your device based on the stent frame. So we've done it, and bench testing was also took place and is absolutely compatible with future access.
Yeah, I imagine for an open procedure, however, that would be much more complicated. For an open surgery, I mean.
Well, clearly is, any bigger valve that you have, like, for example, if you put a supraannular valve like an Evolute in the aorta and you want to take it out, you know better than me that can be a challenge. However, in this initial cohort of patients, one explantation actually had to take place because of misplacement of the valve and the severe residual regurgitation. It admittedly was removed in the 4th month after the intervention, and it was done endoscopically actually, and that was easy to do, relatively easy. It was successful. The valve was replaced with a surgical valve, and the outcome of the patient is very good. So it can happen. Of course, I don't know what it would be like 5 years later, for example, but I think right now we're in the pivotal stage where we're trying to see if this technology is safe, effective compared to historical cohorts. FDA has accepted a single-arm design for all these randomized pivotal trials, because we saw from the SUMMIT trial that randomization of TMVR versus any other therapy is impossible. It's very difficult to recruit patients, and I think if we can see, like with the EVOQUE, that we can have an acceptable safety and very good efficacy in terms of elimination of the MASH regurgitation, this is what we need to find out at the moment. Then we can worry for future problems later.
Okay, thank you very much. We can continue with our next talk.
Okay, so we move from valvular heart disease to coronary disease. The next speaker is Dr. Athanasios Kartalis, who is an interventional cardiologist, director of the cath lab at Chios General Hospital, the only 24/7 primary PCI center in the Aegean region. He's also director of the Coronary and Heart Failure Unit and Pacemaker Center of Chios General Hospital. He has been president of Chios Medical Association. At present, he's a core member of the management board of Chios General Hospital, as well as core member of the evaluation and selection board of cardiologists in the Piraeus and Aegean Islands Health District. The title of the presentation is a classic debate in interventional cardiology, early versus standard invasive strategy in high-risk and STEMI patients. Dr. Kartalis.
Thank you for the introduction. I'd like to thank the organizing committee and especially Professor Patricios for this invitation. The efficacy of immediate reperfusion in the setting of STEMI is well established. When we say STEMI for the students, we mean in the ECG of the patient, there is ST elevation. This is STEMI. The artery is closed, and all the other cases of MI are non-STEMIs. In contrast to STEMI, the optimal timing of invasive coronary arteriography and revasculation in non-STEMI is controversial. The non-STEMI represents a broad spectrum of diagnosis, from normal vessels to multivessel obstructive coronary artery disease. The patients with non-STEMI are classified as very high and intermediate risk. In this talk, we will deal with high-risk patients, which are those with dynamic ST&T changes and grade score higher than 140. The recommendations are clear in patients with very high-risk criteria, because these patients should have an immediate invasive strategy, and which are those patients are those who have dynamic instability, in-hospital arrhythmias, mechanical complications of MI, and recurrent dynamic ST or T-wave changes and acute heart failure due to myocardial ischemia. The things are clear, and in patients with non-STEMI without very high risk or high risk criteria, these patients should have a selective invasive approach. And this is the debate. The patients with high risk criteria, should we do early or standard invasive strategy? And when we say early, we must define what we mean. Is the coronary angiography should be performed within 24 hours from the diagnosis of non-STEMI. Let's see controlled randomized trials, the TEMAC study, more than 3,000 patients. There's no difference in the primary outcome regarding death, MI, or stroke. When we add the refractory ischemia, then the things change. There is a significant difference between the early and late interventional therapy. Another significant study, the verdict, more than 2,000 patients, no difference in the primary endpoint between the two arms, but when we do a pre-specified patient subgroup analysis, the patients with a grade score more than 140, the early phase of therapy was associated with a reduction in major adverse cardiac events. Another study, with fewer than 350 patients. There's a difference between the two arms, but the difference is due, as you see, MI and recurrent ischemia and not due to death. The last study, it was underpowered to detect such a difference because it was terminated early due to slow recruitment due to COVID-19 pandemic. So we have to show the meta-analysis in order to clarify which is the best procedure. The first meta-analysis included 10 trials, more than 6,000 patients, and as you see, there was no difference between the two arms, the early and late invasive strategy, only in length of stay and recurrent ischemia. There was no difference between the two arms in death, MI, and major bleeding. The same findings in another larger meta-analysis with more than 10,000 patients and 17 randomized trials included. The same findings, the risk of recurrent ischemia and length of stay are significantly reduced with early interventional strategy, but the all-cause mortality and MI, there was no difference. Let's see 3 cases. The first case is a man, diabetic, smoker, 80 years old. He had a pain substernal about 20 minutes duration. He had raised cardiac markers, high sensitivity troponin. And let's see the ECG, not something impressive. And we must know to calculate the gray score because, as we said, the patients with gray score greater than 140 need to be treated with early interventional strategy. The gray score, taking in account the age, heart rate, systolic blood pressure, the creatinine, the Killip class, if the patient had cardiac arrest, the cardiac markers, and ST segment deviation. The patient underwent a coronary arteriography. You can see the vessels are calcified. The left anterior descending artery has a TIMI flow 2, and the right coronary artery in the middle has a significant lesion. So this patient has a 3-vessel disease, is diabetic, and the ejection fraction was mildly reduced. The normal ejection fraction is 60%. The heart team decided this patient should have a coronary artery bypass graft therapy. He was programmed 3 days later in a hospital of Athens. The patient was stable and asymptomatic, but in the morning before the program admission, he had a new episode of pain and he underwent an emergent bypass procedure. The second case, a man, 47 years old, smoker with a history of coronary artery disease from his father. He had a substernal pain for 45 minutes. Troponin high sensitivity was high. The gray score was not so high, but you see an impressive finding in the ECG by biphasic T waves in leads V1 to V4. The gray score, as we said, was not so high. And let's see the coronarography. The right coronary artery is clear. There's no stenosis, and you see in the left anterior descending artery a stenosis with a clot formation. The patient had a myocardial infarction, a guiding wire, as you see in the left circumflex, with a stent implantation, a desk stent implantation, 3 millimeter diameter, 23 length, ad hoc angioplasty with a good final result. And the third case, a woman, 71 years old, with dyslipidemia. While she was dancing, he had a chest discomfort with more than 1 hour duration. The high sensitive troponin was raised. You can see in the 2 ECGs the T-wave reversal, and on echocardiogram she had a reduced ejection fraction. This is her gray score. The coronary arteriography did not reveal significant stenosis. So that was a case of MINOCA, means myocardial infarction with not obstructed coronary arteries. The most probable mechanism in this case was considered an atheromatous plaque disruption, rupture or erosion during dancing. Conservative treatment with dual antiplatelet therapy, statin, beta blocker, and ACE inhibitor was applied. This is the most significant slide. An early invasive strategy within 24 hours of diagnosis could be helpful in patients with vessel closure, in whom prompt revascularization may result in salvage of ischemic myocardium. In contrast, a prolonged retreatment with antiplatelets and lipid-lowering drugs could stabilize the plaque and thus optimize the conditions for a subsequent revascularization. The revised recommendation of ESC, ACS guidelines have downregulated the recommendations for early invasive strategy in high-risk non-stemming patients from 1A to 2A, and they recommend the early transfer of high-risk patients with non-stemming to a PCI center. Let's conclude. Non-stemming represents a broad spectrum of diagnosis. A routine invasive strategy for high-risk patients with non-stemming is recommended. However, the optimal timing of a routine invasive strategy remains unclear. Early transfer of high-risk patients with non-stemming PCI center, clinical evaluation, dynamic STT state changes, and PRAISE score more than 140 seem to be useful parameters for choosing early versus standard invasive strategy. An early strategy is safe, does not reduce mortality, but reduce both the rate of recurrent ischemia and hospital length of stay. Thank you very much.
Thank you, Dr. Kartalis, for a very comprehensive talk. Do we have any questions from the audience? Dr. Toudouzas.
Mortality is not reflected or influenced. However, all the other parameters which are really important. The same exactly showed this study funded by the British Cyprus Association Foundation, published in New England in September, for the elderly of 82 years old and more. No difference in the mortality. However, there was some difference in other. So, what's the strategy in your hospital? You are hospital in Chios, in a large island. A patient with non-stemming, 80 years old, comes to your hospital. Which one do you proceed for the invasive or not in your practice?
Yes.
Thank you.
We could proceed to a coronary arteriography. A basic reason is that we have the cath lab. If we were another island without a cath lab, maybe we transfer not all the patients. We could do a selection. So if you have the cath lab inside, I think you should do.
Can I ask you a question, Dr. Gartalis? In the presence of multi-vessel disease, in STEMI, it's probably easier to identify the culprit vessel or the culprit lesion, but in NSTEMI, in the presence of multi-vessel, multi-lesion situation, do you how do you try and identify the culprit? And do you go for full revascularization on the same setting, or do you differ further?
In non-STEMI, you mean?
In non-STEMI.
Yes. We use the ECG and echo in order to help us to find the culprit lesion. And if the patient hasn't taken enough contrast, we take in account the renal function, and if we finished with a culprit lesion in a proper time, early, then we will fix the other lesions in the same time. If we don't, then during his hospitalization, because before his discharge, we'll fix the other lesions.
Thank you.
Thank you very much. We will continue with the final talk of this session. The speaker is Dr. Konstantinos Toutouzas. He's the director of the Cardio-Oncology and Structural Valve Disease Outpatient Clinic of the First Department of Cardiology in Athens School of Medicine. He completed his medical studies at the University of Athens Medical School, his residency at the First Department of Cardiology of the University of Athens in Hippocratian Hospital, and an interventional cardiology fellowship in Centro Cuore Columbus in Milan, Italy. At present, he is a professor of cardiology, director Cardio-Oncology Outpatient Clinic, director of the Valvular Heart Disease Outpatient Clinic at the First Department of Cardiology, Athens Medical School in Hippocratian Hospital, a board member of Aglaia Kyriakou Hospital, and a board member of the Athens Medical School faculty. He is also the chairman of postgraduate program Interventional Cardiology of Athens Medical School. His main clinical interest focuses on the area of interventional cardiology. He performs the full range of interventions in coronary circulation, including coronary angiography, percutaneous angioplasty and intracoronary imaging techniques with intravascular ultrasound and optical coherence tomography. He also successfully deals with the interventional treatment of structural heart diseases. He will be presenting the interventional treatment of septal defects, atrial and ventricular.
Thank you very much, Mr. Chairman. I would like to once more to thank that from the 12th international multi-thematic scientific biomedical congresses, I'm really glad that I was at least to the 10 of them, or 11. I missed one, I think. And I'm really glad I'm here, and I would like to thank the soul of this Congress, Professor Patrikios, for once more for being here. Now, regarding my lecture, I would like to disclose that I'm proctor in several companies regarding these procedures, and I would like to point out, especially to the young students over there, that we have a large family of ischemic strokes, non-lacunar ones, cryptogenic ones, and ESUS ones. Which is ESUS? ESUS, we can define them as the strokes that we don't know the exact etiology of the stroke. This is the major difference from the other kinds of ischemic strokes. And you can see that during a 5-year follow-up, as you may see here, 5-year follow-up, stroke recurrence was almost 30% in patients with ESUS. That means that if you have once a stroke, then in the 5-year follow-up period, you have 30% also chance to have a new one. That means that it's something not stable, but it's dynamic and may change during the progress. And the main cause that we may say that from the ESUS is after that we exclude all other etiologies of ischemic stroke, like atherosclerotic plaques, like valvular disease, like cancer, like left ventricular disease. If somebody, like Dr. Kartalis has showed, if somebody has an acute MI, he may have a thrombus in the LV. So if he has a thrombus in the LV here, in the apex of the LV, this might provoke a stroke. That's a different mechanism. So if you don't have any other cause that may cause the stroke, then we go to the paradoxical embolism. And the main cause is you have some defect between the left side of the heart with the right side of the heart. So if you have a defect here, then you may have a thrombus that may travel from legs, from pulmonary, before it goes to pulmonary circulation, and go to provoke a stroke. The first description of PFO-mediated stroke was, as you may see here, by Dr. Kornheim in 1877. From 1877, in order to treat this PFO-mediated stroke, we went in 1975 when, with this umbrella, they closed this communication between the left and the right atrium. And this is the historical perspective, as you may see here. Since 2008, we have the most of the studies, and after the negative studies in 2012 and 2013, then we had from 2017 and 18 the first positive studies demonstrating that if you close this shunt, you may prevent a new stroke. We have different types of PFO. This is a normal, as you may see here, tunnel anatomy. This is the left atrium. This is the septum primum, and this is the septum secundum. And this one, this membrane here, it's a functional tunnel length that may provoke a communication from the right atrium, the thrombus, traveling to the left atrium and provoking a stroke. The other cause is maybe here aneurysm in the septum primum, which is very usual in these cases. And finally, aneurysmal septum primum that has no stable length of tunnel. This is really something that plays, you may see here in the echo, and this is really dangerous and may provoke a stroke. So what we do to diagnose in order to define stroke, and what we do for the diagnosis of the PFO-associated embolism, we exclude any other plausible causes. If we find one, we stop. If we don't find another plausible cause, then we go for the PFO diagnostic workup, which means that we go for transesophageal echocardiography, and we have the test of the bubbles. We investigate, we have intracardiac shunting. If many bubbles cross this septum, then we have a significant and increased possibility to be a PFO-associated embolism. You're going to see it in the next, in the next image. Here you may see here. This is, as you may see here, this is the left, this is the right atrium, this is the left atrium, and we give from the IV bubbles. And you may see that if many bubbles come from the right atrium to the left atrium, then we say that this PFO is open, as you may see here, and then it needs to be closed in patients that they have already a stroke. Another way to make this bubble test, we can do it by transcranial Doppler. We don't see it by the echo, but we have an echo and we measure the Doppler here in the transcranially. We also give, again, bubbles, and then we measure the score in the, from the transcranial Doppler. Now, as you may see here, we exclude something, and then we say that PFO may be based in foramen ovale. It means that the PFO may be the cause of the stroke. So we need some score in order to have a high chance or intermediate chance or a very low level of chances to have a PFO. The most that it has been used in the literature is the rope score, and you may see the more points you get, the more chance to have a prevalence of PFO. If you have 9 to 10 rope score, then you may see that the estimated the PFO attributable fraction or the prevalence of PFO is 73%. If it's 0 to 3, then it's very low, 23%. So the more of the score, the more chance that you have a PFO-associated stroke. And the points that you get, if you don't have history of hypertension, if you do not have a history of diabetes, if you do not have a history of stroke, if you don't smoke, if there is a cortical infarct of imaging, then you get a point. So if you have hypertension, if you have diabetes mellitus, this means that the chances for the stroke to be associated with the PFO are very low. This is the message that you should get. It's atherosclerotic disease. It's another disease. It's atrial fibrillation, but it's not the PFO-associated stroke. A more descriptive score is also the Pascal score that you may use, which is also the clinical parameters that you showed from the rope score, but includes also anatomical parameters from the transesophageal echo. I don't want to get in many details. This is, as you may see here, very obvious. You can see this is the left atrium, this is the right atrium, and you may see this is an aneurysm from the primum septum that playing between the left and the right atrium. If you do bubbles over there, then easily you're going to see that, as you may see here, the bubbles are coming from the left atrium and quickly going also to the right atrium. That means that there is a defect in this area. Also, the height of the PFO, you may see here, the more the height from the primum to the secundum, the more the height, the more chances that the bubbles will pass from the right atrium towards to the left atrium. So the height of the PFO can also be measured by the transesophageal echo, and also the length of the tunnel. The length was measured also by the transesophageal echo in order to know how to treat this PFO, how to close this PFO. Moreover, if it's very hypermobile, this septum is also the moving and very floppy, then we can also see it by the transesophageal echo. And I already showed you by the bubbles, but you can see it also by the color, by the Doppler color, whether there is communication between the left and the right atrium, especially doing Valsalva maneuver. So if you do the Valsalva maneuver, you may see this color demonstrating the communication between the left and the right atrium. We also can measure other details regarding the intervention, like the angle of the PFO to the right atrium or to the septum. And what are the options now? The options is to give some antiplatelet therapy or anticoagulation therapy, or to close it by an umbrella, a special umbrella. Nowadays, if you have a PFO-associated stroke, you should close it by an umbrella. I will show you immediately how. Technically, it's easy, and there is no problem. This is a survey that we ran in Greece, and you may see that in patients between 18 and 60 years old with a prior PFO-associated stroke, what you consider to close the PFO versus long-term antithrombotic. And 96% of the physicians reported that, yes, you should close. So this is something that's going on. Another significant question, if somebody comes to your office and he'll tell you that he dives, either professionally or he's an amateur and he likes to dive. In this patient, if there is suspicion for decompression illness, again, you should consider to shut the PFO if there is existence of PFO. So something that you have in mind. We have also how to do it, and we have a very nice paper in Intervention 2020. And also another category is patients with thrombophilia. As you may see here, in the old days, thrombophilia was an exclusion to close this shunt, but nowadays it's absolute indication to close the PFO, even with patients with thrombophilia. And this is something we also asked in this survey, And you may see that 85% of the physicians also agreed. Another also parameter that you should get in mind, sometimes if you ask these patients, they complain about long-term migraines. And usually this PFO-associated stroke is associated with migraines. And you may appreciate that after the closure, several times, the patient tells you that he's relieved of this migraine several times. I will pass this in order to demonstrate you a case. This is a case that is really struggling. You may see here that the left disc is in the left atrium, the right disc is in the right atrium, but as you may see here, and I will try to show you again, it's a very mobile septum, and the device is not very stable, so it plays with the septum. So in these cases, we should retract it and stop the case. We're using this Ablazer device, which has the most of the studies in this device, with two umbrellas. This is the Talisman, the new one. We also use this PFO Gore septal device. These are the only devices that have FDA approval, and this is what I'm doing in my practice. And this is a classical standardized procedure. We go from the right side of the heart towards to the left side of the heart. We have a catheter in the pulmonary veins. We appreciate that we are certain now that we are in the pulmonary veins. We can also evaluate it by the transesophageal echo. Then we proceed, and you can see here, we put the device. The left side of the device is in the left atrium. The right is in the right atrium. And after we are 100% sure that the device is stable, then we release it. And this is the final appearance that we have from the umbrella. I'm sorry, I cannot see. I'm struggling with this anyway. And you can see that it's stable. This is the umbrella, the left and the right side, left and right side, and this is stable without any problems. Here is another case. Again, you can see the left and the right side of the device stable before releasing. These are several cases, and I will learn to bother you with more. And we also evaluate, finally, if we have residual sand. You may see here, this is the left, this is the right, and we also evaluate whether there are some small sands between the two umbrellas in this device. We should know it, and we should evaluate this again if there is any problem, because sometimes you may have a new stroke, and you have to evaluate if there is residual sand in these cases. Do we have complications? Generally, it's a very safe procedure, but sometimes you may have complications, as always. Of course, regarding death and all these devices, we don't have any problem. You may have some bleeding from the femoral vein, or you may have some flutter and atrial fibrillation, especially in the first 30 or 60 days after implanting the device. So you have to get this in mind, that you may have atrial fibrillation. It's almost 4 times more in these patients that we do the procedure, but after the 3 months, there is no significant problems regarding the atrial fibrillation, and also sometimes supraventricular tachycardia is a case. Regarding the long-term atrial fibrillation risk, there is was not associated with substantially increased long-term risk of developing AF or flutter. So in the first period, you may have, but later no, will not know. One parameter that we're evaluating, and I'm closing the lecture, is if this device provokes hypersensitivity. If you have an allergy in nitinol, this might provoke more arrhythmias or more problems, or even allergy symptoms. We're evaluating this in this INSPIRE trial, that we do a test before and 3 months after the procedure, an allergy test, and we also randomize these patients to get an Ablazer or a Gore device, and up till now we have watched almost 100 cases. So, in conclusion, the most regarding the closure of these defects, especially with the PFO, is something that we are using in everyday clinical practice. You should know all the workout before going and implanting this device forever, and you should do it selectively. And we have also a school. This is the first, and now we did the second European school in Athens, that we are training people around Europe in order to place the GO device. Thank you very much.
Thank you very much for the excellent talk. Any questions? I think there are several.
Thank you, Professor Toutoutas. It's an excellent presentation. I want to make you a brief question from a pulmonologist view, from a student view that we represent. We have the students here. I want to take a note to the home. Which is the ideal candidate for PFO closure? Is the age? Is the deep stroke?
Yes, I showed you the rope score, and this, I think it's very easy to have it in your phone and you can play with it. But generally speaking, usually 20, 30, 35 years old, women and men, there's no difference in gender. They had several symptoms before, but they don't evaluate them easily. There are some very small ischemic strokes generally. We have seen also large, but generally they are small strokes. Sometimes 20 or 25 years old, they don't even give attention to these symptoms, but generally something is going on. They do the CT, they found the stroke, and then they start also the workout. After excluding atrial fibrillation, which is very rare in 20, 25, 35 years old, after excluding atherosclerotic disease, after excluding any problems with the carotids, then they are doing the TOE, they find that they have this PFO, and then we proceed for the closure. So this is the most frequent workout.
Thank you very much.
Thank you, Dr.
Yes, there's another question from the audience.
Hello, Professor Toutoutas. Congratulations on your presentation. From a student's point of view, I wanted to ask a question. In the case you have an incidental finding in a young patient in his 20s or 30s without a stroke yet, but you find a hypermobile interatrial septum with not a big PFO, let's say, not a very significant one. What would you advise your patient as for example, lifestyle changes?
Yeah, it's a very, very nice question. I would like to translate it more in general. If you find one accidentally, what do? You leave it or not? First of all, a PFO, we can find it. We are here 100 people, around 30, 25, we have a PFO. So it's something very usual. We don't close it unless we have any symptoms or any imaging findings from the brain. Generally, we don't close it. This is the answer. If you have, however, any symptoms, if you have any imaging findings, then you may consider this. But as a primary prevention, this is the question that you asked to me, we don't close it.
There is anything that the patient can do to reduce the risk of a PFO-associated stroke in the future?
You cannot tell. In general, if you see, again, from the history, you find out that they're trying to stand up in the morning sometimes, or they do diving. So you may, but you cannot tell to 25 years old not to stand up very quickly or something. This is something that you cannot do it. I would say we have published, now that I'm talking, about 10 years ago, that in very aneurysmatic septum, you can find also some thrombus very, very rarely. So you may say that you may get some antiplatelets, but this is out of the guidelines. This is something impression that you may have. Dr. Michalis, Professor Michalis, yes. Or Dr. Sinetos, as is also here, very
Hi,
happy today.
If you have a young lady with sort of a PFO, which is one of these PFOs that they have, they are pretty sort of nasty PFOs, aneurysm, tunneling a lot, and then she wants to get pregnant. Would you advise her to close it and have a labor? Would you advise her to have a cesarean?
Without any findings from the...
without any findings?
No, because nowadays, I think nowadays, during pregnancy, they get anticoagulation. So by definition, they get anticoagulation. In all the days that they didn't give anticoagulation, I would consider it. But nowadays, I think...
So you will not advise her for cesarean? You just go have a natural labor?
For cesarean or not?
Yeah.
To avoid, you mean all the Valsalva
high
maneuvers?
pressures, high Valsalvas.
It's out of the guidelines, but I would consider it very, very seriously. Yeah, it's a very nice question and very practical question. Because I was focused on the anticoagulation, but for the cesarean or not, if it's very high risk, aneurysmal, very immobilized, I would consider it.
Now, do you use now, instead of the ROP score, do you use the new Pascal score about the sort of the risk of getting? And the reason why I'm saying that is because the new score has this anatomical sort of characteristics, which usually makes this type of PFOs much more difficult to be closed with an umbrella. So we know that they're high risk, but they're much more different to close, and some of them, they look like the one that you have to abandon the whole procedure.
I fully agree with you, and I think in the clinical practice, we do it. Even if we don't measure the Pascal, we know the ROP score, we do the TOE, and then we know the high-risk features. So practically, we do it, practically, yes. But we write down the ROP score.
Congratulations for the excellent
With
presentation.
Dr. Sinetos, we are doing it together in the cath lab, so Andreas is
what you were talking about is what we published 20 years ago, that is some small thrombi in the atrial septal pouch, which may lead to some embolizations. But this is almost more serious nowadays, that we are going more often to transseptal procedures. So in this case, it's much easier to cause emboli from this septal pouch to go to the brain. But one question for me is that there's some discrepancies between the MRI and the CT finding. So which do you think is the best practice to go if you find an accidental? For example, if you find the CT that it's nothing, if you don't find anything, do you proceed with an MRI or you stop there?
If the symptoms are really suggestive for a stroke, also the neurologists suggest an MRI. So if you don't find anything in the CT and the symptoms are really persuading you that there is something there, MRI should be done, yes. It's a more sensitive imaging method. But I have to emphasize that the question of Dr. Sinetos, usually they come after the workout of neurologists. So they have already the imaging finding.
May I ask a last question about the umbrella fixation of the PFO? In terms of complications, have you ever seen, or is it a general thing to see this umbrella migrating?
Migrating? No. For a PFO, no. Because we have to be really sure that before we leave it, we do the tag test as we do, so we really push and pull. So we know that's very stable. The only case that I retreated it before, the only case was, in this case that I showed you, was really hypermobile, the septum, so I could not leave it. I took it out before leaving it. But it's very, very rare. In ASDs, yes. I remember here also in Cyprus about 7 years ago, it's the only case that we knew it from the beginning, that it was a very large hole over there. We tried to do it, 19-year-old girl. The umbrella left, she went to the surgery, which should be done, or should have been done previously. But she did it, everything is okay. Very rarely, but the PFO, almost never.
Okay. If there are no other questions, there's one in the back still waiting, but really quick if possible. We're already out of time.
Welcome, Professor Grassos. Welcome. Welcome.
Thank you so much. It was a fantastic talk. I'm a neuroradiologist, actually, from Germany, and so we see a lot of patients which are young, and it's devastating because you see them and they wait until they have a stroke because they don't know they will get one. So do you really, like you already answered that there's no enhanced closure for the PFO before they get a stroke, but why would you not, do you really think that the risk of actually closing a PFO is so high in comparison to the damage after a stroke?
Actually, the procedure as a procedure is really very safe. Very, very, very rarely you can see some complications. This is one. The guidelines, according to right now, you should have an imaging method to put a stamp for a stroke, at least in Greece. Otherwise, you don't have the permission to proceed with that, because we proceed. But also the guidelines say that. Of course, the question of 1 million that we had from our student, if you find a PFO accidentally, do you close the primary prevention? Actually, if I found it myself, I would consider it, but I cannot persuade you. There are no studies demonstrating that. On the other hand, I would say, we have a specialist for Professor Naka for heart failure. You see that in the older ages, after 40, 50, 60 years old, PFO is something that reduces the pressure in the left atrium. So it is positive to have a patent foramen ovale, because in cases that you have left ventricular dysfunction, or you may have hypertension, you have diastolic dysfunction, PFO releases the pressure. It decreases the pressure in the left atrium, and this reduction of the preload is beneficial for the symptoms of the patient, and something that is very well appreciated, and especially in patients with heart failure, PFO is something that you need it. And we have now devices, we have a study now with devices that we keep open the PFO. So you have to consider it as from the neurological point of view, but we don't have to shut every PFO because we will pay the price lately in their life.
Okay, got it. Thank you so much.
Thank you.
Okay, thank you very much for the very interesting talk.